Well, good morning again, and thank you for everyone watching. We're going to begin our third session of this program with Dr. Schmidt-Erfurth, who's going to talk about geographic atrophy, identification of disease activity, and therapeutic response by AI tools. Again, for those watching, please put your questions in the chat box, and we'd be happy to answer them at the live Q&A, which is coming up shortly.
Hello. My name is Ursula Schmidt-Erfurth, and as indicated, I will talk to you about geographic atrophy, identification of disease activity, and therapeutic response by AI tools. These are the financial disclosures of all authors. We have all learned with great pleasure that complement inhibition really reduces the expansion of GA lesions. And that insight comes from the FILLY trial, which compared pegcetacoplan-treated eyes in a monthly and then every other month arm with sham treatment. We had the opportunity to look at the images. The FILLY trial evaluation is based on fundus autofluorescence images. Fundus autofluorescence is a 2D method that has some disadvantages. Among others, that it does not easily show early stages, it does not show exudation, and it does, as a method, it is not widely available in the clinic. While 3D imaging using OCT allows an in-depth insight into the neurosensory layers, particularly the photoreceptor layer, but also others like ELM and ONL thinning. So it appears to be an ideal method for evaluation of disease activity and therapeutic response. We have also had to prove in the beginning that fundus autofluorescence, which demonstrates this dark area at the back of the eye, really corresponds to RPE loss on OCT. And by doing this, we reached an R of 0.97, showing an immediate correlation of the loss in the RPE layer in both imaging modalities. So we were confident enough to continue with OCT analysis. OCT analysis includes, in our hands, two layers, RPE loss and photoreceptor loss. So RPE loss uses convolutionary networks as seen here in a schematic drawing, and it provides beautiful en face image segmentation. As I said, the photoreceptor layer can also be demonstrated by 3D OCT imaging using AI analysis. And from a huge manually annotated database and using and validating 4 integrated convolutionary networks, we were able to provide an ensemble-based image segmentation. That means the thickness map of the photoreceptor layer. So what does that look like now? Over time, you see that disease activity, which here refers to the RPE loss, which is shown in red on the left side, you can tell over time how the lesion expands. And you can also tell over time how the lesion expands in the correlation between RPE loss in red and photoreceptor loss, which is shown, and the photoreceptor loss area, which is shown in green. And you can already understand that RPE loss appears to follow the area of previous and pre-existing photoreceptor loss. I will now share with you the tables looking on a direct comparison between the sham arm in light blue, every other month, and all months treatment in pink, in green and pink. And on the right side, you can see the change. So you can consistently see over 2, 6, and 12 months that photoreceptor loss, this is the upper graph, and RPE loss, which is the lower graph, is significantly reduced, particularly by monthly treatment with pegcetacoplan. We could also see that there was significantly less photoreceptor loss, and here come the numbers so that you know that this is highly statistically significant, and that it allows a calculation of the photoreceptor loss area at the level of micrometers. Photoreceptor thinning is another phenomenon that we were able to observe, and that was also significantly reduced under monthly treatment, as seen here, at a highly statistically relevant level. And that was particularly true for photoreceptor thinning in the junctional zone, which was defined as the surrounding 800 micrometers around the RPE loss. So AI-based segmentation of OCT volumes allowed us to automatically detect photoreceptor loss with micrometer accuracy. Moreover, the alteration of the morphology of the photoreceptor layer, whatever that means, just keeping in mind that OCT imaging is not histology, but there's a significant and reproducible change that we defined as thinning, could also be documented. In a post-hoc analysis, patients receiving monthly treatment with APL2 showed a statistically significant reduction in the progression of photoreceptor loss and thinning. So proof of principle that the therapy works. It works particularly, and this proof of principle is particularly based on using AI tools. And this is important as the changes are small, they require high precision, and an automated AI method as a human observer cannot spend hours in delineating micrometer level changes. And photoreceptor loss was shown as the initiating step of geographic atrophy disease. What came out as a major conclusion was also that the ratio of photoreceptor loss versus retinal pigment epithelial loss is the most important predictor of GA growth, as well as therapeutic efficacy. You can see that in the case on the left side, where there is little change as the photoreceptor loss is only is not so far away from the RPE loss, as well as on the right-hand side image. These should be videos, but they do not seem to work. Now it is. So you can clearly tell that the red area RPE loss follows the green area, which is photoreceptor loss. In these images, there was little dynamic in disease activity, so little therapeutic response to be expected. In these images here, there is a huge ratio difference between RPE loss in red and photoreceptor loss in green. On the left side, you can see how the lesion expands towards the optic nerve. And on the right side, you can see how the lesion expands towards the fovea, which is, of course, most worrisome. Here are the numbers. So the 4 quartiles are presented side by side. And you can see that in the largest quartile on the right-hand side, there's the hugest difference in disease activity, as well as the hugest benefit to be expected from treatment. So GA growth in sham eyes increased with higher baseline RPE loss. Sham lesions in the highest quartile showed significantly increased lesion growth, as shown here as high as almost 300 micrometers. And the effect of monthly therapy also increased with a higher ratio. And that was as high as 57%, much higher than the 20% that was found by using fundus autofluorescence in the clinical study. There's also a precise prediction possible based on a baseline scan, and that is very helpful in the future to make an adequate patient selection. So these are the conclusions which I have basically already highlighted, and we hope that OCT imaging together with AI tools will help to identify disease activity, the risk factors for an increased disease activity, which are also the baseline characteristics for a higher response to therapeutic treatment. In this case, it was APL2 that was used, but most likely in other complement inhibitors. AI-based OCT analysis will allow to define robust criteria for patient selection and patient monitoring, and we will certainly be able to develop therapeutic guidelines for clinical trial and real-world disease management, as we are doing in exudative macular diseases by looking at fluid. Automated and objective AI methods is what is needed for an efficient GA management, AI for monitoring, AI for prognosis and treatment guidance. AI technology and clinical management go hand in hand in this case, and I like to thank my team members and also our research partners. Thank you so much for your interest.
Thank you so much, Ursula. That was a wonderful and very insightful, innovative talk, and I look forward to the discussion in a few minutes. Our next speaker is Dr. Neha Anegondi, who will speak on prognostic models in geographic atrophy development and validation. Dr. Anegondi is a clinical imaging scientist at Genentech. Neha.
Hi everyone, I'm Neha Anegondi, a clinical imaging scientist at Genentech, and today I will be talking about prognostic models in geographic atrophy, development and validation. Next slide, please. So these are all the co-authors. Yes, so these are my, these are my financial disclosures. Coming to the introduction, so this is a well-known disease. GA is an advanced form of AMD and a major cause of severe vision impairment in developed countries worldwide. There are no approved therapies to prevent the onset and progression of GA secondary to AMD, indicating significant unmet medical need in GA. Interventional trials focus on slowing the rate of GA lesion growth. There is often a large variability in GA lesion growth rate between patients, which can pose a challenge for interpretation of interventional GA trials, particularly smaller phase 2 studies. So prognostic models for predicting GA lesion growth can enable better clinical trials. So on the right, we see that how the GA lesion grows across time, and change in GA lesion growth rate measured as GA growth is often used as a primary endpoint. And for this, we use lampalizumab studies, which is basically large and long-term clinical trials, which provided us a source of data to build these prognostic models to predict the primary outcome. So prognostic models can be used for covariate adjustment in primary analysis of clinical trials. They can be used in multiple ways: for stratification, for prognostic enrichment, and for covariate adjustment. So what does covariate adjustment do? It increases power by increasing precision of the treatment effect estimate. It removes the impact of imbalance on conditional bias, type 1 error, and power. And there is no risk for the validity of the statistical inference, even if the model is not specified correctly. And on the right, we do see examples of how using prognostic validation, covariate adjustment, you see how precise the treatment effect is as compared to unadjusted analysis. Covariate adjustment is also encouraged by the FDA. Now coming to the study objective, to develop, validate, and compare different models for predicting GA lesion growth rate and to quantify their impact on power of clinical trials. So the modeling endpoint was based on the linear slope of which is the GA lesion growth rate. And the GA lesion growth rate over the typical duration of clinical trials is highly linear, and it can be summarized by the slope of a linear fit for each patient. So it is annualized GA lesion growth rate, which was calculated by fitting a linear regression per patient using the GA lesion area of each time point and then annualizing the estimated slope. So these individually calculated slopes were then used as a prognostic modeling endpoint. And the model can be used to predict GA lesion growth rate at any time point. And you can also use it to predict GA lesion change at any time point. So this is the data strategy, and we used a very clear data strategy which enabled us to rigorously develop all these different prognostic models. So for the training data, we used Proxima A, Chroma, and Spectri. And for the holdout data, we used a part of Chroma and Spectri. And we also had two independent validation data sets, which was Proxima B and Mahalo. So Chroma and Spectri were bilateral GA, and they were in interventional trials. Proxima A was also bilateral GA, but it was an observational trial. Mahalo was also an interventional and bilateral GA trial, but it was phase 2. And then Chroma and Spectri were phase 3. And then you also have Proxima B, which was an observational trial, but the patient population was slightly different as it also included unilateral GA. These are the different model classes which were evaluated. So from left to right, you can see that the complexity of the model increases. So on the extreme left, we have the known prognostic factors, which we also call as the benchmark model, for which we use the GA lesion area, GA contiguity, distance to fovea, and the low luminance deficit. And then we have the pre-treatment GA growth rates, which basically is the benchmark plus a run-in model. And this is a linear model, which is combining the pre-treatment growth rate and the variables in the benchmark model. The third model, which is a work in progress, where we are deriving these features from the mask of the GA lesion using a segmentation algorithm. And then the fourth is the deep learning models, where we are using FAF and OCT images to then predict the GA progression directly from the images. So the model performance was quantified to demonstrate the impact on a clinical trial. So meaningful performance metrics were used to compare these different models and to quantify the expected benefit on a future trial. So models might be associated with different operational or financial complexities, depending on how simple or how complex a model can get. And what we did was we used a simplified and useful starting point, R squared, basically square of Pearson correlation coefficient, as an estimate of the performance. So R squared explains the percentage of variance, and R squared can be translated into more intuitive metrics, such as effective sample size increase, which is defined as shown there. So for example, an R squared of 0.3 means an effective sample size increase of 43%. So covariate adjustment effectively increases the sample size by 43% when compared with a trial using unadjusted analysis. So it basically is like comparing it with a 43% larger trial. So on the holdout set, we did see a very good performance of all the different models, and it was comparable to what we saw in the training set. And as you can see on the forest plot on the left, the FAF model clearly outperformed all the different models that we had developed. And on the right, you also see scatter plots for the benchmark plus run-in model and the FAF model. So the benchmark plus run-in model had an R squared of 0.35, which basically implies a 54% effective sample size increase. And for the deep learning model, it was 0.48. So the sample size increase was even higher there. So the models, and like I said before, the model selected for each model class based on the training data, they show a very stable performance in the holdout data. Now this table here shows how the two independent validation data sets also performed as compared to the holdout. So when you look at just the R squared metric, you do see that the holdout performance was 0.48, Mahalo performance was 0.63, and Proxima B performance was 0.48. So it was very comparable and consistent across the holdout as well as the independent data sets. And when we look at the effective sample size increase versus the unadjusted analysis, you do see that on holdout, Mahalo, and Proxima B, we do see an increase of 92%, 174%, and 91%. And then when you just compare it with the benchmark model, which is basically your 4 prognostic variables, you do see that using deep learning predictions, the difference is still quite large. So you do see an increase in sample size by in the range of 40 to 80% across all these different studies. We do show that prognostic covariate adjustment is pretty powerful based on these numbers. So in conclusion, the FAF-based deep learning model, which used just a single image at baseline, is robust, and it shows a higher performance than models with simple features derived at baseline and even during a run-in phase. And the imaging models can significantly improve the power of clinical trials and decision-making. Specifically, these models can potentially increase the sample size effective sample size by up to 90%, which basically means equivalent to running a 90% larger trial at no additional cost. The future work involves development of additional models, and we'll also talk about ablation studies, which Dr. Daniela Ferrara will also talk in a presentation to explore and identify the image regions which contribute to all these DL predictions. So with this, I would like to thank everyone for listening to the talk and for giving me this opportunity to present.
Thank you so much, Neha. That was wonderful. And it looks like this model is really going to help us going forward in future clinical trials. Our next speaker is Dr. Daniela Ferrara, who is a senior medical director at Genentech and an assistant professor of ophthalmology at Tufts University. And she'll be speaking on deep learning-based ablation studies for feature discovery in geographic atrophy progression. Daniela?
Thank you so much, Dr. Lin, and many thanks to Dr. Singh and Dr. Lang as well for the opportunity to share our work here. And it's my privilege to speak on behalf of my co-authors. And those are my disclosures. So as my colleague, Dr. Anegondi, just highlighted, on the one hand, we're really excited about the potential of advanced analytics and AI-based tools to improve patient outcomes, not only in the clinical trial setting, but in the future in the clinical practice setting. That being said, we understand that the black box dilemma could be impairment for appropriate model adoption, and the end goal is to impair specialists to make informed decisions. So we do believe that exercise like the one I'm going to share today are a valid attempt to bring us more understanding and interpretation of deep learning models. And specifically in the context of geographic atrophy, if we better understand how the clinical interpretation of those deep learning algorithms, we can help us identify or localize new imaging biomarkers that can lead us to new insights to the biology of the disease, and ultimately help us develop trust that the deep learning models' outputs are worth acting upon. Our group and many other groups have been endeavoring such efforts to better understand the deep learning algorithms and which pixels those models are really seeing in the image. And our group and several other independent groups have been using approaches such as attribution methods. Even though they're quite insightful in some circumstances, one disadvantage is that attribution maps can fluctuate even between models with similar performance. And therefore, it's very difficult to really understand how good the attribution maps can be in specific circumstances. For that reason, the exercise I'm going to share today, we use the ablation methodology as our preferred method. So a good advantage is that allow us for direct derivation of where the prediction signal is coming from, but we should keep in mind that relies on annotation masks performed either by humans or by other models, and that should be taken into consideration in the exercise I'm going to discuss. So specifically to understand and better bring clinical interpretability to the GA progression prediction models that Dr. Anegondi just discussed, we picked our GA fundus autofluorescence images, and we selected 3 regions of clinical relevance. The first one was the GA lesion itself, so inside the lesion. The second one was the GA rim, and the third one was the image background beyond the image rim. And I'm going to discuss how we played our models, take into consideration all those regions. Before I go to the exercise, I want to mention that we used for this project almost 1,300 eyes, and they were breaking training data sets into 5 folds. And we used cross-validation to train 5 models. So the exercise I'm going to describe now, we either kept the regions of interest or we ablated the regions of interest. And we compared how the model performance was impacted in comparison to the model trained in the totality of the images. So in the first exercise, using the 3 regions of interest, we ablated either the GA lesion, the rim, or the background. And what we learned from this exercise is that ablating the rim decreased the performance of the models in most experiments in comparison to the full image. As a retina specialist, I was relieved and reassured that the rim in that exercise was the most relevant image region. And to confirm that observation, we did the other way around, and we retained only the pixels from the 3 regions of interest and then compare with the full image again. And in confirmation, the best performance came from the models using the rim region information. And then we asked ourselves, what importance is really coming from the shape and the size? Because natural history studies, and there's extensive literature on the topic based on human annotation, shows us that lesion shape and size at baseline are predicted to GA progression over time. So when we use the masks only without the pixel texture or intensity information, what we observed is that information, those features of shape and size, they can explain roughly a third of the model performance. So still relevant, but not the entire progression prediction information is coming from shape and size alone. And then as a final exercise, we ask ourselves, what's the relevance of the intensity information? And to address that scientific question, we used a very simple but interesting methodology. We simply shuffled the pixels of the image regions of interest that I just described. And what we observed is that shuffling the pixels from the regions of interest that I just described, when we compare the rim with texture and the rim without texture, that really makes a difference. And when we compare the background with texture and the background without texture, that also makes a difference. So that tells us that the intensity information is not that relevant, but the texture information is truly what's relevant. And again, as a clinician, I felt reassured, because when we look into fundus autofluorescence images, we really don't pay attention to image intensity. We do pay attention to fundus autofluorescence patterns as extensively described in the literature. So in conclusion, this exercise shows that the GA rim appears to contain the most important region features that our deep learning algorithm used to make such strong progression predictions at baseline that Dr. Anegondi just described. And we confirmed that the shape and size are relevant for that prediction, but they don't explain the entire variability. We also observed from this exercise that fundus autofluorescence intensity, really what counts is the fundus autofluorescence pattern, but it doesn't seem to be relevant to the intensity of the pixel brightness itself. So 2 main future directions coming from both works that we shared today. Even though those exercises cannot pinpoint exactly the imaging biomarkers that give us the most important features, we know that they are close to the GA rim. And again, that can help us to better understand, identify future biomarkers that could be used in future clinical trials as endpoints. It can shed additional light into disease pathogenesis and understanding. And we also understand that similar efforts should be encouraged to help us better understand the deep learning technology and ultimately make sure that this technology is truly empowering experts, whether scientists in the clinical research or whether physicians in the clinical practice. The technology should empower experts to make informed decisions. Thank you very much.
Thank you so much, Daniela. That was a very clever project for deciphering the areas of the autofluorescence that are involved, and I look forward to our discussion. Our next speaker is Dr. Gagan Kalra. Dr. Kalra is a research fellow at the Cleveland Clinic and also founder of IT. He'll be speaking on utilizing machine learning for automated segmentation of geographic atrophy lesions on SD-OCT. Gagan?
Thank you for the generous introduction, Dr. Lim. It's a pleasure to be here, and I'm going to talk about automated segmentation for geographic atrophy lesions, as was said. Okay. I'd like to acknowledge some of my co-authors who've really made this project come together, and I'm going to head straight into the I don't have any straight financial disclosures, and there are some from a team that's been listed here. So geographic atrophy is a late-stage finding in age-related macular degeneration, and it's characterized by retinal outer layer and RPE thinning. This can be visualized on optical coherence tomography, as was highlighted in some of the talks earlier. And it can also be characterized with hypertransmission defects into the choroid, which can be visualized on fast OCT images as hypertransmission features or window defects. The traditional gold standard for GA measurements has been fundus autofluorescence imaging, but the widespread use of OCT in the new CAM consensus definitions has laid a renewed emphasis on OCT imaging in geographic atrophy monitoring and otherwise in clinics. So as was highlighted earlier, machine learning has been used for several different aspects in ophthalmology. It can be used for automated segmentation, detection of disease, or even prediction and prognostication for several of our disease entities. The current analysis that I'm sharing today is based on utilizing machine learning to localize and segment geographic atrophy lesions precisely with pixel level area measurements and presence or absence like binary detection using standard near infrared OCT images and cross-sectional B scans. We used 900 spectral domain OCT scans from about 341 subjects with dry AMD with or without geographic atrophy. This data came from Heidelberg Spectralis device or the Zeiss Cirrus device. Those are the two devices that most of the images came from. And an automated machine learning enabled multi-layer segmentation tool, which has been previously validated, was utilized to segment the ellipsoid zone, the Brooks membrane, and the RPE, which can be visualized in this figure on the right side. So then a triple level expert grader review was also conducted to make sure that the ground truth segmentation was really the ground truth and any segmentation errors were corrected. Geographic atrophy training masks were then automatically generated based on cross-sectional locations where EZ, RPE, and Brooks membrane overlap with each other. And that was the definition of GA used in this study. And identified masks were then compiled to generate an on-fast mask as well as a cross-sectional training mask. So that way we were then able to train two different models. One that was just solely based on B scans and was looking at B scans as input images and GA masks on B scans as training masks, and on-fast based model, which used the compilation of those masks over the whole volume and looked at an on-fast raw image as an input. This was fed into a UNet architecture, and the details for what the parameters were for the training of the UNet are also listed on the slide. The model performance was then additionally compared between the two device types that were included in the study, just to see how those generalized across different, scanner types. So going to the results of the study a little bit, the B-scan machine learning model for localization and segmentation of geographic atrophy yielded a high accuracy of 94% with a sensitivity of 90% and specificity also of 90%. The model performance appeared to be lower for Cirrus type scans compared to Spectralis scans, although the performance was not that different. In the figure, you can also see the ROC curve for this model, along with the sensitivity and specificity that's also listed on the figure. Moving on to the results from the on-fast based model. This model yielded a higher accuracy of 0.96 with an F-score of 0.82, a sensitivity of 95%, specificity of 93%. And again, bear in mind that these metrics are from pixel-wise measurements of GA area. And this is just not just the presence or absence of GA, but precisely measuring where in the volume exactly GA is. And the comparative device performance was very similar between the two devices as well. So that goes to the credit to the model that, the data set was diverse enough that it's able to perform well, regardless of what device the images came from. And the figure here shows the ROC curve. Okay. So this slide shows some examples of the outputs from our B-scan model. The first panel on the left, you would notice has a raw image. That's the figure, this label as A, the raw B scan, and The figure B shows basically what the ground truth annotation was for identification of C-RORA or geographic atrophy. And in the figure C, you can see the blue areas are the areas where the model and the ground truth overlap with each other. And the figure D shows the grayscale output of confidence of the model of exactly where it thinks GA is in that scan. So it seems to do a pretty good job of identifying where exactly it's marking, and it's very explainable, as well as some of the talks before have highlighted the use of masks in training, and instead of using just the black box, you have more explainable results than where exactly is the model looking for features. The second panel in the middle then shows an example where in a disease state where the RPE atrophy is present, but there are areas in the scan where the RPE atrophy is not complete. This is something that we call iRORA with the new CAM definitions, so the incomplete retinal outer layer and RPE atrophy. So the areas labeled in orange in the panel, the middle panel, figure G, if you notice, are the areas where the false positives were seen, and essentially it's marking the areas where the RPE is still pretty disorganized, but it's not completely absent, as was in the definition of the model training. And the confidence map, or the deep learning grayscale output, reflects that the areas which are very bright are where the model was very confident as where GA should be marked. And the last panel that's on the right is it shows an example which has a pretty high pathology. There's some RPE deflections that appear to interfere with how the model would comprehend this image, but it seems to still hold up pretty well. Areas which are very small are labeled as false negatives in some of those areas. So if you notice in the right panel, figure K, the areas which still appear green are areas which were ground truth labeled to be GA, but the model seemed to undermark that a little bit. And the deep learning grayscale reflects that also. And this next panel shows a set of on-FOSS images and how our model performed with that. Essentially, it does an incredible job of identifying areas that it thinks are marked as GA. And the deep learning confidence, the pattern of the images on this slide as well follows the pattern from the previous slide. So the left panel shows a typical scan, how that would perform in the segmentation. And the panel in the middle shows that the on-FOSS images sometimes come with its own set of challenges, that there may be aberrations in the image, or just the reflectivity of the tissue might vary, and it might not be the window defect that's coming from absence of RPE. And the model tends to see that and mark that as false positive. And in the right panel, you may notice that in scans which have artifacts, or you can see that there are horizontal aberrations in the image where the GA lesion itself is not as contrasty and easy to identify. The model tends to undermark a little bit in such images. So it really boils down to the quality of the input images that goes into the study, and you are able to achieve higher performance. So I'd like to conclude by saying that a high performance on-FOSS and B-scan based model was achieved with a relatively small data set. And this model performance generalized well across both devices that were tested. Automated quantification of GA enables size-based lesion thresholding as well that can fit into the iRORA, C-RORA distinction based on size. Future endeavors in this area are already underway, where we are validating these findings in external data sets. I'd like to acknowledge my team at the Coli Clinic and under the leadership of Dr. Ehlers and the Ophthalmic AI Summit for this opportunity.
Thank you, Gagan, for showing us that NFOS-OCT can achieve the same outputs as other models. So we look forward to that discussion as well. Our next speaker will be Dr. Frank Cheng, who is the president and CCO of INUC, and he'll be speaking on Ophthalmic AI. Why is it far ahead of other healthcare AI applications? Frank?
Thank you, Jenny. So my topic is, why is ophthalmology AI far ahead of other healthcare AI applications? As we all know, AI applications have been mushrooming in every field of medicine. This is especially true in diagnostic-related fields, such as radiology, cardiology, ophthalmology, and pathology. The topic I'd like to explore today is which healthcare segment is actually leading that AI movement. On the surface, obviously, radiology and cardiology segments are more visible. They lead a number of FDA approvals and clearances for AI. Specifically, in this recently published list by the FDA for nearly 350 AI and machine learning enabled medical devices approved by the FDA as of mid of last year. Actually, 70% of the listed approvals are in the field of radiology, followed by cardiology of 12% and 3% for hematology and neurology. So the number of ophthalmology AI clearances is actually far behind in the number on the chart. However, if we focus on the degree of innovation and the scale of transformation, many believe that ophthalmology AI is actually leading the AI movement. Among these believers, Eric Top was one of them, as evidenced by his tweet about a few years ago. Here on this slide, I'm contrasting some obvious differences between the best-in-class ophthalmology AI and the radiology AI. First, on the degree of automation, actually 2 ophthalmology AI technologies had already been cleared by the FDA. That is the IDXDR technology and the IR technology. And none of the radiology AI technologies actually had reached this level of automation cleared by the FDA. Further, when it comes to clinical performance, the FDA-cleared ophthalmology AI technologies have been rigorously tested in our field against ETDRS. Well, on the other hand, American College of Radiology and RSNA, which is the Radiology Society of North America, sent their joint letter to the FDA last year, stated there's pretty significant concern. They stated that autonomous AI in medical imaging or radiology could actually be significant patient safety concerns. And finally, when it comes to payer reimbursement, the new CPT code in the ophthalmology field, 9229 here in the US, is now widely covered by payers in the US. On the other hand, the leading radiology AI is only covered by Medicare for hospital inpatient service and not for radiologist work. So what actually enabled the advantages that ophthalmology AI now enjoys over other fields? In my view, there are several important reasons. The first reason being that have been the ophthalmology field benefits from some of the pretty well-established clinical consensus on disease definition and the scale. This is especially true in DR diagnosis, which is supported by both ICDR scale as well as the ETDRS standards. And the second reason being, I can attest from INOC's standpoint that ophthalmology AI companies actually benefited years ago from some of the early support and thought-leading guidance from both the National Institute of Health and the National Eye Institute. So take my company, INOC, as an example. We received our first grant from the National Institute of Health more than 10 years ago. And since then, $10 million grant have been granted to the company by both NIH as well as NEI. All of these grants enable INOC to now become one of NIH's national small business success stories. And the third reason being, ophthalmology AI innovations would not have been adopted so quickly, in my opinion, without the strong support of organizations such as AAO and ASRS. In this example letter to a regional contractor of Medicare here in the US, when AAO was influencing the payer to cover autonomous AI technology for DR screening, they stated, this new retina imaging procedure allows early detection of sight-threatening DR with immediate point-of-care feedback to the patient. And they went on to say it promised a significant advance in public health and substantial program savings by reducing the need for costly treatment of advanced DR disease. Right? So now that obviously that was very supportive. If we take that AO support letter for granted, on the next slide, I'm showing you a very different letter from a different field, right? So in the radiology field, American College of Radiology and RSNA sent a very different kind of letter last year to the FDA. In their comments, right, these two organizations stated that autonomous AI in their field, which is radiology, could bring significant patient safety concerns. So hopefully you now understand and agree with me. There's some pretty structural support and the reasons behind ophthalmology AI, which probably led to the industry-wide success. So now I'd like to just to give you a quick overview of our industry, ophthalmology AI industry's kind of quick landscape. Actually, AI is active in all ophthalmology segments, especially in DR, AMD, glaucoma, and also ROP. Among them, there's no doubt autonomous AI segment for DR detection is the most active and visible segment, with 2 FDA clearances to date already, and the CMS national reimbursement here in the US. Now, obviously, there is a number of players, but I'm going to just use our IR product as an example to show you how this works. Right? So the IR workflow is very simple. It enables generalists to conduct diabetes eye testing at settings such as primary care and the diabetes care. In those settings, any operator with a high school diploma can be trained to use the fundus camera and submit images to the IR AI cloud. Diagnostic results are typically available within 30 seconds after imaging, and typically no dilation is needed for most patients. In addition to DR, AI, there's a lot of brands and the companies out there in the field. In addition to IR, over here, due to time, I'm not going to name every company and also go into their details. But what I like to do is just to share a few key considerations when you select a autonomous AI for DR detection. So I would, propose, right, key considerations when selecting autonomous AI would ideally include the following 6 criteria. First, published results for AI accuracy. Second, the extent of the regulatory approvals in key markets around the world. 3 is the depth of diagnostic details. And 4th, whether the algorithm has been subject to any head-to-head performance comparison with published results. And 5th is the extent of the worldwide adoption. And finally, government insurance company, reimbursement coverage. Right? So I'm going to just, use a few examples from the sample and IR to illustrate these points. Okay. First of all, a diagnostic technology's accuracy, regardless of in ophthalmology or beyond, right, needs to be rigorously validated. For example, IR has been extensively validated around the world. Recently, JAMA Network Open published this paper with results from IR's prospective 15-center clinical trial. In this trial, IR achieved 96% sensitivity for more than mild DR and 97% sensitivity for vision-threatening DR. And more importantly, 88% of the patients were able to be diagnosed by IR without dilation. So how was this study done? Right. So, the IR results was based on undilated 2-field fundus images by novice operators, which is then compared against the ETDRS reference standard generated by leading US reading centers, expert readers. They use obviously stereoscopic 4 wide field images acquired with dilation. Now, moving on, the second consideration I proposed earlier when selecting AI technology is actually the extent of the regulatory approval in key markets. For example, this is IR's FDA, approved indication for use, right? So, which was achieved back in August of 2020. FDA basically allowed IR to automatically detect more than mild DR and vision-threatening DR for each patient eye. This approved indication use makes IR the only approved, FDA-cleared autonomous AI technology able to report results at such specific diagnostic details. Fourthly, head-to-head AI comparison results. Over the years, IR has participated in 3 head-to-head comparison studies globally, each with published results, and IR performs well in each of them. Specifically, IR was the best performing algorithm in the UK study about 5 years ago, which was published on Ophthalmology. IR was also the best performing algorithm in the US VA study published by Diabetes Care in 2021. And in recent months, UK's National Screening Committee published their results of their 10 algorithm review. And their conclusion was that IR is the only AI technology ready for live clinical use in UK's screening program. So the final consideration is, obviously the extent of worldwide adoption. Here in the US, since FDA clearance, IR operations has grown to more than a dozen states. The current users of IR includes hospitals and health systems, eye care practices, primary care and diabetes care clinics, federally qualified health centers, and service companies for HEDIS gap closure. Overseas, outside the US, we've been supporting paying users of 14 countries. IR is the only autonomous AI technology approved in major markets such as the United Kingdom for live clinical use, Canada, and Saudi Arabia. In the market of South Africa, the largest medical insurance company there, which is called Discovery Health, recently announced that they chose IR as the first reimbursed AI product for their patients' DR screening. And actually in Germany, 3 out of the 4 best diabetes hospitals ranked by Newsweek are using IR as we speak. Okay. Now, last but not least, insurance support, both from government and private insurance, and also clinical society's acceptance are critical for AI technologies adoption. We're happy to report that autonomous AI technologies approved by the FDA here in the US are well supported by all key clinical institutions, which includes AAO, AMA, ADA, ATA, as well as NCQA. Okay, so in summary, based on specific evidence I provided earlier, I believe that ophthalmology AI is far ahead of other fields, and it is ready for real-world adoption now and poised to become standard of care very, very soon. So with that, here's my contact details, and I really appreciate the opportunity to speak, and we'll be happy to answer questions later.
Thanks so much, Frank, for sharing with us why AI is so far advanced in ophthalmology compared to other fields. We'll move on now to our next speaker, Dr. Daniel Ting. Dr. Ting is the current associate professor in ophthalmology with Duke at the National University in Singapore, the Singapore Medical School. And he'll be speaking on 5 rights of AI in ophthalmology. Daniel?
Thanks, Jenny. Thanks for the kind invitation to speak. Hi from Singapore, everyone. For the next 10 minutes, I believe, the earlier panelist has given a great overview about the AI use case, why AI is ahead of any other medical expertise. But I just also wanted to actually touch upon some basic principle in terms of how we should actually be understanding and as well as implementing AI in the real world settings. So this is some of my financial disclosure. So we know COVID has hit us hard over the past 2.5 years, and that has kind of resulted in the urgent need for digital transformation worldwide. And the impact that COVID has resulted in the global healthcare landscape include the increased public and staff exposure risk. We see the increased waiting time for the patients in the hospital system. Some patients unfortunately experience a delay of the treatment, and partially also because of the healthcare workers were deployed to the front lines as well. So when COVID first hit, we were thinking whether is there any role for AI or other types of digital technologies to help mitigate some of the risk for COVID positive patients and non-COVID positive patients. Together with Larry Kareen, who's the Vice Provost, the President of Duke University, then Victor Zhao, the current President of National Academy of Medicine, and Tian Wong, we have actually jointly published this paper in March 2020. That was just 3 months after the outbreak has started in China. Basically, it was actually used as a reference piece by the US CDC. So basically, in the paper itself, we have actually broadly divided the AI and digital innovation technology into 4 big categories, both for the COVID positive patients and for the patients with no COVID infectivity. And some of the examples that we've included in the papers include Johns Hopkins COVID-19 dashboard, how AI has been used to predict COVID-19 spread worldwide, and some of the medical imaging analysis using the deep learning techniques on CT scans and chest X-rays as well. So if you actually look at the industry revolutions, right now we are currently at the 4th, and I would even dare to say that we're currently at the 4.5. So we've been through STEAM, electricity, computing, and AI, machine learning, and deep learning is currently where we are at. And AI is nothing new. It started back in 1950s, and machine learning term has been coined by Arthur Samuel back in 1980s. And back then, he's already said that the computer should have the ability to learn without being explicitly programmed, right? And this has not come to fruition because of the lack of the supercomputers. And of course, Jeff Hinton has first described the deep learning technique back in the late 80s, but it didn't come to fruition as well until like the advent of the GPUs that has sparked off the whole entire deep learning era starting from 2010. And if you look at the deep learning, these are the 3 big godfathers you have to know: Jeff Hinton, Yoshua Bengio, Yann LeCun, and jointly they published these reference papers. And this is what I would call it as a classic landmark papers in deep learning that's been published in Nature, that's currently been cited more than 40,000 times. It says that the deep learning should have the ability to learn and should have multiple processing layers that analyze the data with multiple levels of abstraction. And this has heavily adopted in many different domains, includes speech, the image recognition, and natural language processing as well. So if you break down CNN into the 3 big different layers, it could be broadly divided into the input layer, the hidden layer, and the output layer as well. So most of the CNN differs based on the hidden layers as well. If you look at the evolution of the CNN layers over the past 30 years, it has actually increased from 7 layers, as first described by Yann LeCun, LeNet, all the way to like 2016, the DenseNet. You can actually see the ResNet even push beyond 1,000 layers. So I mean, those are the layers, the evolution of the CNN that's been happening over the past 30 years. So if you look at the non-medical domains, such as the AlphaGo, the gaming industry, so AlphaGo team has actually published this paper first in 2015, and then followed by 2017, AlphaGo 0. So using this deep reinforcement learning technique, the AlphaGo team has spun off another team called AlphaFold team, and they have actually successfully used the similar techniques to predict the protein, how the protein can fold and unfold. So if you actually ask a human to actually manually calculate how the protein can fold and unfold, it takes us 13.8 billion years. It's impossible. So using the deep learning, the machine can actually do it. And what is the implication in the medical healthcare settings? Potentially, we can use this technique to develop therapeutic targets, or we can potentially even modify how the disease, can progress or can stop the disease from progressing. If you look at the deep learning applications in the healthcare settings, has been widely adopted to CT scan, the ECG, the skin, as well as the retina imaging, as many have actually earlier alluded to. And started in 2015 and 2016, we have actually started the deep learning journey. So we published a journey together with, the AI experts groups worldwide, including Lily Peng, Aaron, and many others peers. And we have actually published this paper in VJO and PRER, summarizing all the state-of-art AI technology many years ago. And we've actually did a follow-up paper to see how AI could actually be integrated, and not only just a standalone technology, but how does it actually talk and connect with other digital technology. So I mean, in the PRER paper that we published, we actually touch upon some of the newer technologies such as 5G, 6G, IoTs, the emerging digital technologies like home monitoring devices, AR, VR, and how these emerging technologies can actually sync with AI applications in ophthalmology. So, I mean, just coming to my talk, the 5 rights, I mean, I'm just trying to actually summarize the principle into 5 different categories. First, you need to ask the right questions. And in order to build a good AI algorithm, you need to use the right data, the right partner with the right concept. And then, of course, you need a right enablers to actually implement this to the clinical settings. And what is the right question? So in fact, what is the unmet need? I think this is usually the first, the question, the intended use, right? And what is the market size we are currently thinking about? And of course, I mean, for the Singaporeans, we always say like, how do we use it outside Singapore? Same thing for the US developer. How do you actually use it outside US and vice versa in other parts of the world? And of course, you always have to do like the literature review and how does your technology pitch against the other technology that's been described already in the market. And diabetes is a good market size. That's why, hence many companies are actually developing this. If you actually look at the, the unmet need, by 2040, 1 in 10 people will have diabetes worldwide, and this is a huge market to be addressed. And if you look at the right data, I mean, rubbish in, rubbish out phenomenon is something that we should always avoid, and I can't emphasize this point anymore. So basically, if you actually were to access the data, so different hospitals, different countries will have different regulations. How do you anonymize the data? How do you clean them? And what the size are we talking about? The build and robust AI algorithm? What is the phenotype of the data that we are actually need in order to not just develop, how to validate and even test. And right technical team, I think of all the 5 points, I think this is the lowest, lowest hanging fruit in my mind, because I think if you actually handle the other 4 well, I think the third is actually, identifying the technical team using the right technique is actually the simplest of all 5. And I think like between the clinical and technical teams, I think the rigorous communication is just like a marriage, right? I mean, husband and wife always have to talk. Same thing between the clinical and technical team. You always have to communicate. And about, clinical team, you always have to define again, unmet need, what's the medical terminologies. You have to always explain what's your clinical workflow to the technical the colleagues, and whereas the technical team has to actually, explain what the technical methodology, as well as how to get the sense about the clinical data sets and the ground truth as well from the clinical team. And the right concepts itself. So how do you build like the AI architecture? I think this is where the clinician can come in very in a very important sense. So, I mean, how do you design like it's no different how you actually design the hospital settings, like where do you want the patients to go? Where's the waiting room going to be? How are you going to dilate the patient? How are you going to image the patient? Same thing in the AI system. When the image comes in, how do you pre-process your image within your AI system? Do you recognize the right versus the left? Is it a chest X-ray versus a retina imaging? And how do you actually perform like data augmentation, the contrast enhancement, and then followed by feeding into the CNN? And then of course, if you want to do like a post-hoc analysis to explain what your AI is saying, and this is some of the things that you have to actually include that in your AI system. And right enablers, I think I always say this as a value of depth. How do you, after you have a research AI algorithms, how do you actually bring it to the market? How do you productize the AI algorithms, and how do you actually obtain regulatory approval? I think these are all the important steps that one actually should know. So global AI trend, I mean, this is something that I've actually learned over the past few years, standardizing of the reporting guidelines. How do you actually handle the data privacy and data sharing issues, the cybersecurity, generalizability, the last mile implementation? How do you explain about what the AI is seeing, and medical, legal, and of course the AI biases and the ethics as well. So just a very brief slides on privacy preserving technology. This is one of the methods that many the technical teams are using. How do you share data without needing to send the data physically across the border? And the other technology that I would like to share is the blockchain technology. It's a distributed ledger technology. It decentralizes the control so that you can achieve a trusted peer-to-peer transactions. It bypasses the need for central authority, always begins with a genesis block, and the transactions are stored within a fixed size block. And each block will always be linked to the previous one to actually form an endless chain. So if you look at the unique characteristic of blockchain, is immutable, is decentralized, is secure, and is auditable as well. And blockchain has been heavily adopted in the COVID setting as well, to actually look at vaccination status, the COVID testing status as well, to make sure that the patient's identity are being preserved. And we have actually published this paper in the Lancet Digital Health, looking at how blockchain can be used to improve the AI trust as well. So 5G, 6G, and telehealth is something that I've actually mentioned before in the previous slides. And quantum computing, super exciting technology that would potentially push, I think in my mind, the fourth industrial revolution to the fifth, and how do we actually, evolve from the GPUs to the quantum computers. I think there's a lot of research that's actually being invested into this field. So if you think like a supercomputer GPU is super powerful, and a quantum computer can take 3.5 minutes to do something that a classic GPU can need to take in 10,000 years. So this is something that quantum computer potentially can actually bring up the protein folding to the next level. And then metaverse, I mean, it's something that's heavily been discussed about to humanize the interpersonal, the connection, the human interaction. Is this going to be a future? I mean, this is all the super exciting future that we're talking about. With this, I would like to thank the organizer for the kind invitation to speak. Thank you.
Thank you so much, Daniel. That was a great overview of what we need to do to get AI right. I'd like now to open the panel discussion and have all my panelists unmute themselves, please. And we will begin. So there have been a lot of interest here from the group. And I'd like to ask first Dr. Ferrara. There's a question. You know, the rim you showed was very significant, right, as to predicting progression. The question is, were there certain types of patterns in the rim that you saw that could lead to faster rates of progression that we could pick out?
That's a great question. And this is part of our upcoming experiments. So again, one of the main goals of this exercise was exactly to better understand specific image biomarkers that would be related to GA progression. And I want to also acknowledge a parallel effort that our group is conducting using OCT. And I think multimodal assessment will also be very insightful. So short answer is not from this exercise, but this is actually next steps for the group. So biomarker identification based on that region.
Great. And Neha, a question for you then. Building on what Daniela has responded to, you know, using your model, you're able to decrease the N, right? Because you're able to give sort of like an increased sample size. Going forward, are these going to be implemented in clinical trials? And is it really possible to do this just from one image? I find it fascinating.
Yeah, thank you. I think that's a great question. And then regarding whether we are thinking of implementing it clinical trials, so those are the discussions we are having right now with the different teams within Genentech to try and understand if they would be open to implementing these models. Because like we said, it's free lunch. There's no loss in using these models and trying to understand how it could help in understanding the treatment effect. So yeah, that's definitely an ongoing thing.
And Jenny, if I can add to that, I think what several speakers alluded to this, we have to decide where to implement this technology. And if we think about the patient journey, either the patient journey in the clinical trial setting or the patient journey in the clinical practice setting, we know there are milestones and bottlenecks where the unmet need can be addressed by digital innovation and AI-based tools. So for example, patient screening could be one very relevant use case, and even clinical trial design, stratification factors could be another use case. We are actually starting from the end. We're starting to consider implementation of those tools in the data analysis because that's the safest. You don't impact your clinical trial conduct or patient safety, but you can leverage AI to the clinical trial interpretation. Once this technology is well established and we are confident on the understanding and utilization of AI at the end, then we can start moving towards earlier phases of clinical trial implementation.
Thank you. And Daniel, you've had extensive experience using AI. Have you gone into the clinical trials area in Singapore? I know you have Siri in Singapore that actually reads images for multiple diseases already and gives diagnoses.
Yeah, well, I mean, thanks, Jenny. I mean, for Singapore, we have not actually gone into the clinical trials as yet. I mean, in terms of using AI to help read, the say for instance, AMD, DME, or like a GA, for instance. I think like I am not aware of like the any US FDA approved OCT related the AI algorithms as yet that's been approved. I mean, and in Siri alone, we have done the DR screening technology, but so far it's not in the clinical trials settings as yet. Yeah.
Thank you. And Frank, you talked about AI in ophthalmology. Is there any new development using AI in your company for AMD, say? Because, in AMD, we have defined stages akin to, like in diabetic retinopathy. And so I think that would lend itself nicely, perhaps, to an AI system for AMD diagnosis.
Sure, Jenny, good question. So yeah, we have always been very clear about INEC's goal and role in doing more than just DR, right? So we have been researching deeply autonomous AI for years related to both AMD and glaucoma, right? So both our algorithm for AMD and glaucoma has, transitioned from R&D stage to beyond R&D stage. And that's all I can say at this point. So hopefully we will make some announcement for non-US markets pretty soon related to our algorithm for AMD and glaucoma.
We'll be waiting to hear that. Gagan, there's a question regarding the ground truth. You were talking about how the segmentation looks at what's there and then what's absent. And the question was, if there's loss of photoreceptor RPE and the choriocapillaris, how were you able to segment that? Like, how did the algorithm know which layers were missing?
So I emphasize that the ground truth generation was using a semi-automated ML-based algorithm that has been previously validated. Basically, we wanted to be sure that the ground truth that was going into the model training for identification of GA was really the ground truth. And there were several layers of expert reader review that was involved. And when there were mistakes that the automatic algorithm would make, that's where the semi-automated aspect of it in correcting the ground truth would come into picture. And this is something that's been used in clinical trials extensively in the literature as well. And basically, when the layers are absent, the way it does is like in complete atrophy or C-RORA, the thing that you're seeing as the only thing that you can visualize on OCT is like the Brooks membrane, and the RPE and the outer layers are kind of absent. So the way that is segmented is that those will be overlapping the Brooks membrane.
I see, thank you. There was a question for Dr. Erfurth, but I believe she had to leave. And the question was, how does photoreceptor loss, does it always precede RPE loss? I just want to point out that in the Pinnacle study, which is a prospective study looking at imaging in GA, that they actually did see greater percentages of photoreceptor losses that preceded the RPE. So it goes nicely with what Dr. Schmidt-Erfurth had presented, showing the photoreceptor to RPE ratio. For any of you who are left on the call, would you want to tackle the question as, why does photoreceptor loss precede RPE loss?
Well, I don't think any of us has an answer, right? And I think there are two main schools in the field, if you will. Some of us believe the photoreceptors loss is the beginning, and we think about AMD and GA as a neurodegenerative disease. Others think of AMD and GA as a lack of choriocapillaris and think the whole pathological process starts from underneath. And I think the field agrees that what we see dying the latest is RPE, and there's extensive data in the literature showing that. So, and it's possible that the photoreceptors loss and the choriocapillary changes are actually happening in parallel, and it's possible inflammation and oxidative stress are playing a role. So I don't think any of us would have an answer, but there's strong body of evidence showing that photoreceptors are one of the earliest levels of the tissue to suffer.
Yes, and I remember Jean Dewan used to say that AMD is really a photoreceptor disease. And so perhaps he was right. Maybe, where it all starts. And, I find it interesting too that, in our anti-VEGF treated eyes, that frequently there's already RPE loss also occurring as the photoreceptors die. And then even if we try to treat with the, anti-VEGFs and we get rid of the CNVM, that subsequently we do get RPE atrophy. And that it's there very early on in a significant proportion of patients, as was shown, even in the AREDS data set, even when they look back at Harbor, and that it was not related and not caused by anti-VEGF. So I think, that's kind of important that we all, realize that. So I applaud all the work that you are all doing, to try to decipher what layers are missing. And just a question, I'm very curious about this algorithm that you've developed looking at, the rim and looking at the texture. Does the texture actually get to the heart of like what we see clinically as this trickling pattern or this corona pattern versus nummular pattern? Can you address that in the last few seconds here?
We believe so. So we do believe that FAF patterns are relevant, but I do want to leave you with a concept that we touched very lightly here, which is, and Daniel alluded to this, which is the data set that's used to build the algorithms in the first place. And in our exercises, we used data from the lampalizumab program that had an inclusion-exclusion criteria. So not necessarily our data sets are encompassing the entire phenotypes of GA. And I think this should be top of mind for anyone using AI, understand the limitations of a broader generalization of our outputs. So yes, we found FAF patterns, but they were related to the population that was used in the first place.
Thank you all so much. You're all experts in the field, and thank you for sharing with me and with the audience today. Have a great rest of the day, and we'll move on now to session 4. Session 4 will attack more of the practical aspects of pearls and pitfalls and what's necessary to transition into AI for the clinical applications. And our first speaker will be Dr. Jayashree Kalpathy-Kramer. She is the chief of the new division of Artificial Medical Intelligence and Ophthalmology at the University of the Colorado School of Medicine, the Anschutz Rogers Eye Center. Prior to that, she was at Harvard, where she was in the radiology department as a full professor. Dr. Kalpathy-Kramer will be speaking on a practical guide to pitfalls and the applications of machine learning in medical imaging.
Thank you so much for having me here today and for the opportunity to share some of our experiences. As was mentioned, my background has been a lot in radiology and other imaging fields. Here are my disclosures. In addition to these disclosures, and we have received funding from many different entities, including NIH, NSF, internal funding, but also some from Genentech and others. So in addition to the disclosures, I also have some disclaimers. I am not an ophthalmologist, so sort of stating that up front. Although I've done a lot of work in ROP with Dr. Michael Cheng and Pete Campbell and others for more than a decade now, a lot of the other work we've been doing has been in other domains, including radiology and oncology. And although we heard just previously how ophthalmology is ahead of radiology in terms of where it is in the hype cycle, maybe that's a debate we can have another time as to the validity of that assertion. And based on our experiences in all of these different domains, I agree there's a lot of reason for optimism and excitement about what AI and machine learning can do for us here. But also based on our experiences in these other domains, as well as in ophthalmology, I have to say that there's also some reasons for concern, and we should be cautious and aware of these before we sort of very broadly deploy these algorithms. Yes, I just recently moved and very excited about this new opportunity to spend more time in ophthalmology. So we've seen all day today, and we'll continue to see for the rest of today, that it is getting relatively easy to create AI algorithms. Almost nobody here is starting writing their algorithms from scratch. We all use frameworks such as TensorFlow and PyTorch and all of these things. We have access to GPUs, and the combination of these open source algorithms and frameworks, along with access to large amounts of data and the hardware today, this confluence of factors has led us to a place where we actually are in a relatively good position to create these AI algorithms. We've seen some phenomenal work from peers and others where clinicians have been able to, without much coding, create these algorithms that are really very good. And so we are really getting to the place where essentially you take your annotations, you take your data, you take something like AutoML, you put it all together and out pops an algorithm, and it works quite well. You have another algorithm, for instance, that does NLP, and it can write your abstract for you. It might even write your grant for you. We're not far from that today. But despite all of that, I think, I guess I would argue that it is, despite all of these advances, it is still not trivial to create algorithms that are not brittle, that are sort of self-aware and know when they may be wrong, that are explainable, and that are unbiased and fair. So I would like to just sort of touch upon some of these, again, based on our experiences, both in ophthalmology, but also more broadly in many of these other domains. So one challenge that we've seen reported is the brittleness of these algorithms. You have a data set, they work really well, you train and test and do validation within that data set, and you get phenomenal results. But the minute you try to do external validation, whether it's a different imaging device or a different population, all of a sudden you see that the algorithms have been quite brittle. And we've actually seen this reported in the radiology literature fairly extensively. And there's a lot of reasons for data heterogeneity that can lead to poor model performance on these external data sets. We've also seen about how the FDA has been evaluating these data sets. And one of the limitations that people have reported, at least in the radiology field, is that even though many of these algorithms have gone through the approval process for regulatory aspects, very few of them still have done extensive external validation. Very few of them have done prospective studies. And so there is still a reason to sort of more closely examine how these algorithms will work in the wild. And we've seen that reported in ophthalmology as well. As, the algorithms that was so wonderful in one setting really did not work all that well when they're deployed in a completely different setting. So some of the challenges that we see are different populations, different ethnicities. We've heard this in terms of even something as much as sort of trivial as of the fundus, the colors of the images, the ethnicities, things like that can make the image appear quite different. And there are many ways to mitigate against that, but that is something we have to check for. One thing we notice is people typically report the behavior of the algorithm on the entire population and don't talk about it, how well it works on subpopulations, especially minority vulnerable subpopulations. And that is something as a community that we need to be really, really vigilant about, is that just reporting it on the entire population is not a good indication for how well it's going to work for everyone. We see different camera systems. They might have different fields of view. The pigmentation, I mean, the color might be different. The illumination might be different. The sort of things like texture that are hard to appreciate might be somewhat different. And we saw this extensively in other fields, like in cervical cancer screening, where even though the humans couldn't quite tell what the difference was, the algorithm was very sure that this came from this camera system and this image came from this other camera system, and behaved quite differently in these different protocols and what kind of images you might acquire. So we were expecting to see fundus photographs and we suddenly saw this. And the algorithm thought this was a fundus image and then happily gave a prediction on this image, but that is not what it was trained on. And so obviously that it didn't make sense to try to do that. Image quality is a huge issue, and we need to make sure that the algorithms have some kind of filter potentially on input validation in terms of, is it the right training data, and different biology. So in terms of these common pitfalls, COVID was mentioned previously, and we see a huge amount of work that came out during the COVID era. And the summary of many of these review papers is that none of these papers are worth deploying. And there were hundreds and hundreds and hundreds of them. And most of them is because of some of these brittleness issues on these sort of Frankenstein data sets and things that people didn't really examine. These algorithms reported excellent performance, but are completely not usable. The heterogeneity, whether it's different prevalence of disease, different scanner systems, and more complicated is the association between disease and some other confounder. So whether it's things like obesity or age or other things that are sort of confounding the association between the factor of interest and other demographics is something that we don't talk about enough again. So here's data from radiology, but you can see when you train and test on the on-diagonal elements, you get great performance. On the off-diagonal elements, meaning you train on one system and test another, performance just falls off. And so if you had access to all of these, The other thing I would like to say is that what we find repeatedly is that these algorithms first learn the device, and then within the device, they learn the disease, which means they really cannot generalize to an unseen device very easily. Because now if you have a new device, it'll be a fourth cluster, and it's never seen that, so it wouldn't know how to treat that device properly. So obviously there's been many attempts, and we greatly appreciate all that NIH has done to create these multi-institutional data sets for across domains. And if we had access to such data sets, we can get great performance, again, only within the populations that were represented in these data sets and don't necessarily generalize externally. So there's many reasons that is a challenging. Patient privacy is obviously a primary one. There's a need for harmonization of metadata. People are very worried about holding on to their data for a variety of reasons. And logistically and practically, sort of the expense required to create a well-curated, anonymous public data set is not trivial, and very few researchers have the ability to do that. So one way in which people get around that is things like federated learning, and we are very excited about the potential for federated learning. The idea here is you can learn from multi-institutional data sets without the need for data to be aggregated, but rather model sharing, and there's many different ways to do that. And essentially that allows you to learn from a corpus of decentralized data. And there's many different approaches. Very most common one is something that does federated averaging. So you sort of give the model to each of these clients, they train locally, they update it to a central server, you aggregate that, you send an updated model back, and this process continues till you have a good model. And we sort of did that for ROP and showed that it works really well. And it's really important in cases like diseases that are low, rare diseases where you don't have sufficient data at each institution to train a model. You put it all together and you get a really good model. The other problem is they can be black boxes, and we saw different ways of dealing with that. We've seen grad camps used very commonly. They're like all over the place, but they really are fundamentally flawed. And we've shown in radiology that they can really are not repeatable, they're not reproducible, and trying to put sort of clinical assessment to these is fraught. And there's been a ton of work saying, be careful when you use these kind of methods for explainability. And I was very very excited to see some of the other methods that had been talked about previously in terms of occlusion and other ways of looking at explainability that are not post hoc. Another thing is these algorithms can fail silently, and you won't know that they failed. And we have no idea of knowing which case is the algorithm failing on. So the whole process is sort of uncertain. And so we need to think carefully about how do we sort of get around the uncertainty issues. And so there's ways using sort of Monte Carlo deep learning networks that give good assessment without having ground truth that the model is working well, or the cases where the model is likely to be bad. And then race is another issue. Bias is another issue that was talked about. Most of the data comes from certain parts of the US, and we really need a diverse data set. And AI has superhuman capabilities. So between the not being transparent, the risk of bias, and superhuman, we really need to be very careful. So this sort of final thoughts is very excited about what AI can do, but there's many, many different challenges, including the need for diverse data sets, the risk of bias, fairness and bias need to be fully established before we can deploy them. So thank you.
Thank you so much, Jayashri. That was a great, insightful presentation of all the pitfalls to temper our enthusiasm for AI. And our next talk may address some of those pitfalls that you raised. Our next speaker will be Dr. Michael Cheng, who is the director of the National Eye Institute and the NIH. He's an expert in ROP, AI, and bioinformatics, and previously was at OSHU as a full professor. And Dr. Cheng will be speaking on how can we support AI research, the National Eye Institute perspective. Michael?
Well, thank you very much for the opportunity to speak. I'm Michael Cheng, Director of the National Eye Institute, and I'll be speaking from the National Eye Institute's perspective about how we can support research in artificial intelligence. And if you're interested in what we're doing at the National Eye Institute, follow us on Twitter at NEI Director. And we're really interested in trying to post information about things like grant opportunities and just news of interest to the community. So I will just begin by talking about what we do at the National Eye Institute. We were founded in 1968 by Congress and then President Lyndon Johnson as a branch of the NIH to manage national efforts in vision science. Okay, our budget this year is $863 million. And we're really proud that the work that we've done has had really broad impact on science overall, and that's really clear in artificial intelligence, because so much of what's been innovative in AI has really begun in the vision field. Work of Michael Abramoff's and many others. And we 6 months ago published a new strategic plan. If you go to this QR code, it'll take you to that plan on the NEI's website. And in short, there are 7 of what we call cross-cutting areas of emphasis, and data science is one of them. And I just want to highlight a few things that are relevant to this meeting here. The reason that this is one of our areas of emphasis is that there is just unprecedented access to large-scale data, and that really helps define 21st century science. And one of the advantages of the ophthalmology field is that regardless of what type of data people are interested in, from biological data to imaging data to clinical data to population health data, it's actually fairly readily available in the vision science field, compared to almost any other field. And so there's this core set of data science methodologies that we use to analyze that data. Now, what are the unanswered research questions? Well, these have been covered by many of the talks here, but if I could just summarize a few of them. One of them is that the performance of AI systems tends to get better as the task that they're asked to do narrows. These systems are very good at, for example, rule out macular edema, rule out plus disease and retinopathy of prematurity, but the real world is just more complicated. It's multiple diseases and findings in the same patient. And it's not just imaging data, you've got to integrate image data together with clinical data. So a lot of challenges here involving integrating systems and really coming up with clinically relevant use cases. Another challenge is generalizability and bias. Okay, the real world is not just homogeneous data sets. It's different races, different populations, different imaging devices. And you in general cannot just take results from one population and extrapolate to the others. The real world data is often much poorer quality than research data. A lot of biases in data collection, and only 6% of deep learning studies had any external validation based on this study in the Korean radiology literature. And I got this slide from Jayshree Kalpathy-Kramer, and now at the University of Colorado. And how do we deal with this? Larger data sets, more diverse data sets, and really validation across different populations and different devices. And as has been talked about at length here, another challenge is unclear ground truth. There's often diagnostic variability because of rater heterogeneity and just gray zones. We often have very unclear definitions of clinically significant endpoints. How do you define moderate glaucoma, severe glaucoma, rapidly progressive glaucoma? And do you look at the optic disc? Do you look at the OCT? Do you look at the visual fields? And what are the cut points that you use? Different experts will use a lot of variability here. And so we've got a lot of work to do involving things like consensus definitions and how do you develop those reference standards or ground truth. So what are we trying to do about this, and what do we feel like we need at the from the National Eye Institute perspective? Well, one of them is data sharing and harmonization. Again, modern research really relies on big data, and one of the challenges is data sharing. Okay, we often have this paradigm where there are different centers around the country that are basically working on the exact same problem in parallel. But each of those studies is individually underpowered to really answer that question. And so, the investigators often are not incentivized to share data. And in fact, even if they were, they generally can't share anyway, because they each collect their data under different data representations. And so it just takes too long to harmonize. And so in terms of data sharing, yes, there is an NIH data sharing policy that goes into effect next year. And that's a stick. Okay, you've got to come up with a plan. Otherwise, we don't fund your research. I really feel that we need more incentives for data sharing, more carrots. And about a year ago, we worked with Marco Zarbin, with Pierce Keen, and with Aaron Lee, who are Marco's the editor-in-chief, and Pierce and Aaron are on the editorial board of TVST, the ARVO Open Access Journal, and created a new publication type, a vision science data set or software library. And the point of this is to try to gradually shift that culture to really incentivize data sharing and team science. Okay, you can get academic credit for publishing your data. People will cite it, and you find it using PubMed. And I think we're going to need more and more of those carrots. And we're going to need common data model for ophthalmology. And who's working on that now? There's an AAO group together in collaboration with some of us at the NEI. Sally Baxter at UC San Diego, Carrie Getz at the National Eye Institute, and Michelle Rybar from Oregon are some of the leads on that project. And so data sharing and harmonization to really address some of these challenges. Another thing that we need is image standards. Okay, so much ophthalmic data comes from devices that really generate pictures and numbers. Okay, this is a gold mine for research involving artificial intelligence. Okay, all of you know this. And one of the challenges is that it's often very, very tough to get data out of these devices because they're locked in proprietary standards. Okay, I personally worked with an informatics PhD student in Oregon who wanted to get OCT and visual field data to study machine learning and glaucoma, couldn't do it after a year, changed to a different project. And we see this all the time. And we at the NEI have been working with the ONC and with the FDA to try to solve this problem. Okay, we've been doing this for about 9 months now, meeting really, really regularly with them. Now, the ONC, led by Mickey Tripathi, and why them? Because they manage the Office of the National Coordinator, sort of interoperability and electronic health record, electronic data exchange. Why the FDA and their group in imaging devices led by Malvina Edelman, because they regulate devices. Okay, why us? Because we manage grant funding. And so we've been working on what are those levers we can use together as federal agencies to really promote widespread adoption and standards adoption for interoperability. We had a workshop about a month ago, and we're really trying to work on this. And more information about this coming out soon. Yeah. One of the other things that we need is larger data sets that are AI ready. Okay, a couple of things I want to let you know about. Our Bridge to AI is a common fund project. Okay, it goes across all of the NIH. And we were one of 5 institutes that really led the development of this project. And almost all the research that we traditionally do at the NIH is hypothesis driven. This is different. Bridge to AI is $100 million over 4 years for hypothesis generating research. It's basically building data sets, okay, that are AI ready. We hopefully will have information about the winners to announce about this fairly soon. Aim Ahead is another project that's trans NIH. And the premise of this is that we're using artificial intelligence to address challenges of health disparities and minority health by developing diverse data sets, training a diverse workforce, and developing AI approaches to detect bias in medical records. And I really hope that objective image data can play a big role in this. Again, this is going to be a big project, $100 million over 4 to 5 years. Okay, so NIH projects involving larger data sets that are AI ready. Another thing that we need is medical education in AI and data science. Okay, what is the one tool that every single clinician and scientist uses in this country? It's the computer. And despite that, there's basically no systematic training about the usage, how do you use these technologies? Okay, compare that to taking a history, doing a physical exam, okay, auscultating the heart. Okay, the average student or resident spends months to years of dedicated training on that. Yet for the one device that we use all the time, there's basically zero focused training on that. And the other paradigm is that the older person teaches the younger person in medicine, but in this case, it's the older person who tends to know less. Okay, so we've got to turn that upside down. Okay, the other challenge is finally clinical documentation in EHRs. If this is going to be the source of data that we use for AI clinically, well, we did some studies, okay, 3 million EHR notes, analyzed them with Adam Rule and Michelle Rybar in Oregon. Okay, these notes are long, 6 pages on the average for ophthalmology, heavily redundant. 75% copy pasted or templated from a previous note, and it's getting worse over time, over 10 years. And so that really makes us question what's the research value of these data. So finally, we're in a world where we're moving from qualitative to quantitative. Okay, that's true for AI, medical imaging. And the question is, well, what's the added value of clinicians if these machines all generate numbers and everybody sees the same numbers, regardless of whether you are a retina specialist or general ophthalmologist or optometrist or patient using Google search. And so I think we've got a lot to figure out. How do we understand these technologies and just maintain those uniquely human skills that may end up being that added value? So thank you very much. A real privilege to be able to speak here today.
Thank you so much, Michael, and for leading the initiatives on trying to tackle some of these problems with AI and data sharing. Our next speaker is Dr. Luis D. Cisternas, who is a scientist at Carl Zeiss Meditech, and he'll be speaking on the AI bias. How do we set performance expectations? Luis?
Thank you, Dr. Lee, for the introduction, and thank you, everyone, for the organizers for giving me the opportunity to speak with you today. So I'm going to talk about AI bias, and we have heard about it before, so hopefully I won't be too repetitive. And I'm also going to talk a little bit about what kind of tools can we use to determine the uncertainty of these models and reduce this bias. Okay, so I'm an employee at ZEISS, but I have to say that this representation slide deck is my own, and it's not does not represent the company. And I am the lead of the AI clinical decision support team at ZEISS, and also I'm showing this on behalf of my collaborators at ZEISS and all these type of institutions that I have here that have made this presentation possible. So we are currently at stage that we are developing a lot of different tools for either the staging of disease, and here's an example of AMD staging tools that we are developing, and also for prognosis on disease that can tell us what is the best treatment for each patient and how we're going to treat each of these patients. So there is like a lot of different tools that we're developing. And one thing that we have seen in common for all of these tools is that what do we need to make them work is data of good quality. So one of the things is like what data is going to work for these tools. We can develop some quality control algorithms that will tell us, even they can be in artificial intelligence that will tell us which data is appropriate and which data has enough quality. So is that all that we need? So if we just need data of good quality to make this work, our presentation will end here and everybody will be happy, and that's all. But that is not the truth. We have heard throughout throughout today that there is a bias in AI, and the bias comes from the data you have been training with. It's also the bias that you're training with and the bias that you evaluate your model with. So we see all this great performance of these models in papers, and then you go to real life and they don't really apply. And more importantly, in different populations as well. So we have, just by experience, we have models that we have trained for fluid segmentation in a European data set and then tested European data set, and you have very good accuracy. And then you move on to a different data set, like the Asian data set, and they don't work as properly. So all these numbers that we see in the publications look really good, but what happens to that extra 10 or 5% where they don't work? It is important because if it consistently failing for the same population or same patient characteristics, then we have a big problem. It is not just, it's just 5%. It's just like there's a big problem. It's consistently failing for particular populations. There's also different problems that we have that the imaging devices keep evolving continuously. So we cannot have a model right now that we can be sure that it's going to work in the future. And we cannot just rely on image quality to give us an idea that the model will work or will not work. And I just wanted to give an example of this bias, just something that we train with OCT and geography. We train one of those denoising algorithms that try to find the vessels a little bit clearer. And we had the superficial and deep retinal slabs. And we saw on top here is the before the algorithm is applied, on the bottom after the algorithm is applied. Incredible result. The noise was eliminated. Everything was great. And then we got a choroid capillaries slab. And this model had only seen the superficial and deep retinal slabs. When we applied to the choroid capillaries, we saw that it was creating something completely fake, creating really small vessels on the choroid capillaries. And we know from the literature that it should not look like that. It should look more like punch holes in a net. We added some of these choroid capillaries data to the model, and we had something that was slightly better, but we still cannot, we don't know for sure if the small vessels that appear there are real vessels or just bias from the data that we had trained the model with. And in fact, if we use a lot of pictures of cats, we can come up with the choroid capillaries looks like a cat, which is absolutely not real. And I don't know if you guys have used PowerPoint before or are using PowerPoint for that, but there is this feature now that as you drag images into your PowerPoint, tells you what they are automatically. And obviously, none of this PowerPoint has seen any OCTA images, so the automatic description that comes out for all of these images are completely varied and nothing to do with angiography. And the one for the cat is correct because it has seen all the pictures of cats. So what do we have here? We cannot just trust image quality and that's it as a metric to evaluate whether our models will work or not. We need something that will tell us implicitly, based on the data that the model has seen, whether it will work or not for that data. And this kind of comes like a third category in our model that says, I don't know what this data is, because then we have like a dog chasing its tail, trying to figure out it will have its own bias on having this I don't know category. So we need to have a way to really tell the model based on the data it has been trained. I don't know how to deal with this. And that's something that we're trying. And I just wanted to give you an example of some experiment that we tried. And we tried to apply an idea like this to segmentation of geographic atrophy in OCT. So first of all, we generate these pseudo-color images, which are based in different properties of characteristic of geographic atrophy in OCT. And we just train a model that use these pseudo-color images and manual annotations. And we decided to use a five-fold cross-validation. So that means that we are dividing that in 5 folds. We use 4 folds to train, and we use 1 fold to test this model. And we do this 5 times, right? To train this model, we use different augmentation techniques in which we can rotate, translate the images, and also change the color, the pseudo-color images, how they look like depending on the intensity that we kind of simulate that the OCT signal has. So then afterwards, we have trained these 5 different models. So we have a separate data set, and we can evaluate these models that we have trained in a completely independent data set. And we had 2 different graders to provide manual annotations for this comparison. So what did we find out? That is that since we have 5 models, we have this 5-fold correlation, we have 5 models. And these 5 models were identical network, identical number of epochs, identical the training parameters, everything was the same. But we saw that as we passed the same testing, independent test image to these 5 different models, we obtained different results because there is something different there. The only source of variance that we had there was slightly the data they had been trained with, as they were part of the 5 different folds. So we thought like this, since the only thing that we vary is data, this has to tell us something at the epistemic uncertainty, or what is the unseen manifestations that the model has never seen, and what is the, so any difference that we find between these results, it means that it belongs to the data that the model has been trained with. And then we can also perform augmentation at our test stage, and we perform different augmentation depending on the signal, the simulation of the OCT signal level, and we also see different results. So all the results that we see difference give us a sort of idea of what the model is, what kind of signal levels the model is familiar with. We put it all this together, and we can have an average of all of these results, and we can have the standard deviation of the results. From the average, we can threshold, or we can try to outline what is the predicted geographic atrophy location, and that's our segmentation results. The standard deviation gives us an idea of uncertainty. Just give us an idea of what data the model has never seen in terms of the data population and in terms of the different intensity levels of our data. We apply this for to a number of cases, and we, for example, this very easy to delineate case, we can have here the segmentation result pretty accurate. The uncertainty, it always goes around the different border of the geographic atrophy, because that's the harder part for the model to do. We have another example here in which the is more heterogeneous, the atrophy, and we can see highlighting as uncertainty. The model is telling us, I don't really know what this is. I don't really know if this is really geographic atrophy of its background in the exact same place where there is more heterogeneity, because our model was not trained with enough of these heterogeneous cases. And then we have another case that has some areas of heterogeneity and some image artifacts, that straight line that you see in the bottom half of the image. Again, the model is telling us, I don't know what's happening here. This area, I'm not sure. It can be deemed as uncertain. We can use these uncertainty maps to threshold what we consider where the model is correct, absolutely correct, and where we consider where the model has these different difficulties. And we can set different thresholds about what we consider the model to be certain or uncertain. What we discover is that the regions where there was agreement between the model and the readers have lower values of uncertainty. Those areas were always very certain. The regions where there was a disagreement between model and the 2 readers, there were higher levels of uncertainty, which was a good thing. It's kind of showing that these uncertain levels really mean something. We started masking different regions of uncertainty depending on the level, and we saw that as we masked more and more and more regions, changing the uncertainty level that we consider acceptable, we had better and better and better performance to our models compared to our readers. The performance between the 2 readers changing these uncertainty levels did not change whatsoever. So that was telling us that it's not the difficulty of the task outlining the region by the readers, but more implicit to the model, telling us where is uncertain and where is not uncertain. What happens is that if you start throwing away data, you end up with no data. So something that we make sure of is that we can set a level which we have the same performance on the 2 graders, and we also only saw throwing away 5% of this data. This will be very useful, for example, for reading centers. They can run this model, figure it out, which data sets are 95% will be good, completely certain, good to go. The other 5% are the ones that we'll have to look into and try to edit the segmentation or get a better result. So I just want to conclude that we just don't need images of good quality. We also need a way to tell which regions are certain and uncertain. And the best way to reduce all uncertainty is to have more and more and more data. So I agree with the previous presenters today that we need something to get together and get all the data possible. And one of the ways is Federated Learning.
Thank you so much, Luis. That was great and really illustrated the need to eliminate bias, to have big data set, and to know what the machine doesn't know. So our next speaker now will move on to Dr. Michael Abramoff. Dr. Abramoff is the founder and executive chairman at Digital Diagnostics, and essentially the father of the first autonomous DR machine out there. And he'll be speaking on the adoption of rigorously validated AI to enhance access for patients from point of care to specialist care. Dr. Abramoff is also the Robert C. Watsky Professor of Ophthalmology at the University of Iowa. Michael?
Hi there, my name is Michael Abramoff. Very excited to be speaking to you. Sorry I couldn't do it live, but I will be live at the panel a few hours from now. The title is long, but essentially about adoption, how important rigorous validation is, what the role of ethics are to move those patients that need care by an ophthalmologist or retina specialist from the point of care, diabetes care. I'm at Iowa. I'm also the chair of the Collaborative Community in Ophthalmic Imaging Foundational Principles of AI Workgroup, and I'm founder and executive chairman of Digital Diagnostics. I have obvious conflicts of interest, which are listed on the bottom of this slide. I want to make sure we understand this word autonomous versus assistive AI. So assistive AI means that the medical decision is made by the clinician. Liability is also with the clinician. It also means that the patient already is in workflow. They're already there with the doctor. The AI just helps make a better, faster, cheaper diagnosis. The real world value is in hopefully improving outcomes for patients. Autonomous AI is very different than you see an example system, IDXDR. In fact, the first ever FDA authorized autonomous AI system in any field of medicine. There, the medical decision is made by the computer. The liability is with the AI creator. Therefore, you can do it point of care and immediate result is available. And it can be added wherever a patient already is. It doesn't have to be with a specialist physician. Real world value also means that you can not only improve outcomes for patients, but also for entire populations, including improving access and addressing health disparities, which is a big item in the last few years. This all started with an ethical framework. And so rather than explaining it in my usual way, we started with an ethical framework, which was published, widely discussed, that led to things like how do we solve for liability, and leading to the American Medical Association AI policy in 2019. On the left, you see how from this ethical framework, we have been creating regulatory frameworks, at least considerations. We'll talk about the paper next. And it also led to inclusion in the standards of medical care in diabetes from the American Diabetes Association, a patient organization. So that's an important achievement for getting this to patients and hopefully improve health disparities, which was the goal, improve outcomes and health disparities. On the right, you see also how we developed a payment framework, not only how we can get this reimbursed, but also how can you as an AI creator put a value on the AI per patient or per population. So the paper is coming out, actually I heard 2 days from now in Nature Digital Medicine, very exciting. Very proud of that paper with my collaborators. It also led to care gap closure with an autonomous AI. For the first time, HEDIS and MIPS language supported the use of autonomous AI to close a care gap, thanks to a lot of work by the National Committee of Quality Assurance, or ACQA. Then next, with the AMA, the DEMPEC, and the CPT editorial panel, the first ever CPT code for an autonomous AI, which you probably know as 92229 in 2019, and ultimately ending up in nationwide Medicare payment of about $55 to about $80 blended rate from private payers in this year, starting this year. I mentioned the ethical framework and that this all started with that, not with an algorithm, but how do you embed this in the healthcare system in the right way. That led to this paper I mentioned together with FDA and the Federal Trade Commission, We created these considerations, we call them, everything from how you conceptualize an AI to how you develop it, how you train it, what the input and outputs, how you can update it, what a reference standard is. There's no time for me to go into detail, but there's a lot of information in there about how to build AI the right way. And there's multiple other publications by this work group. We're very proud of this first one. And then I mentioned already what's coming out 2 days from now in Nature Digital Medicine, a reimbursement framework for AI payments, starting with the AI creator. How do you actually put a value on the AI and its function, all the way to payers, but all other stakeholders, and how their involvement allows you to do AI the right way in healthcare and bring these patients from the point of care to the specialty care when they need it, when they need management or our treatment. And so this is crucial in my view. We need to have all stakeholders accept, because they have questions such as, is it safe? Does it improve outcomes? Will it worsen health disparities? Is there racial or ethnic bias? What happens to my data, and who's liable for errors? We have addressed all of these in these frameworks, and that was necessary to make sure that all stakeholders, payers, physicians, patient organizations, bioethicists, regulators, all accept AI as solving a major problem in the right way. I want to say a little bit about this ethical system. Typically, when you develop an ethical framework, you start with ethical principles. You may have heard them in medical school or elsewhere. Such things as patient benefit or no maleficence, first do no harm. Another ethical principles, justice or equity, where we want people to be equally treated. It's more complicated than that, but that's one way of thinking about it. Autonomy, where the patient can make decisions about their own healthcare. And finally, responsibility on the bottom right, leading to liability and medical malpractice considerations. So the point of this framework is not so much to put nice words onto AI, but more allow these things to be measured in a way that an AI engineer building an AI creator, coding the software, choosing the training data can actually measure how much each ethical principle is being met for patient benefit, for equity and bias, for responsibility, and for patient autonomy. And so what you do in the AI system in yellow is here in the middle is try to optimize a balance between the ethical framework doesn't tell you where to put the balance, but it does allow you to choose how much you want to factor in each of these different ethical principles, and how do you find the right balance. It's not prescriptive, it's especially a system of metrics. So all of this led, and already mentioned it, inclusion in the standard of medical care for diabetes, which is part of the American Diabetes Association standards work that many ophthalmologists are involved in. I already mentioned autonomous AI can close a MIPS or HEDIS care gap now. The language specifically says that an AI can be used at, not only a human. And then finally, very important movement, thanks to the support from the American Medical Association, especially the American Academy of Ophthalmology, but also the National Hispanic Medical Association and National Medical Association, all work together to create this national payment for CPT code 9229 at a rate that makes sense from a business perspective for an AI creator, and in a blended rate, what we see given private payers of about $79 to $80. So I sketched this path now in a different way. You start on the left with an algorithm, ethics are prime, that leads to, if you do it right, FDA regulatory approval, solving for medical malpractice and liability, standards of care, making sure that it fits care gap closure for MIPS and HEDIS, reimbursement, CPT coding. You need a framework for creating AI payments there, so it's not a one-time off. Ultimately, why are we doing this? To improve health outcomes, improve population health outcomes, and preventing blindness and visual loss in our patients. And I think that's happening. A few data points is we work very closely with Orbis in a partnership where we try to expand this FDA rigorously validated AI to many countries, including Bangladesh, Saudi Arabia, other countries, South Africa, several countries in Europe. So that's exciting. And also in the US, rapidly expanding. We cannot keep up with hiring people to implement the systems that we sign up. And all these states, of course, have coverage and payment. So that's really important for the system is adopting it to make sure that they are financially rewarded for doing the right thing, which is getting their patients diabetic eye exams. Finally, 2 examples, one in New Orleans, inner city. New Orleans, there was diabetes care, but no eye care left. Hundreds of patients didn't get the diabetic eye exams. We came in with the AI. Now, all of them got their diabetic eye exam within a few months, thereby preventing visual loss and blindness. And another in FQHC, which is very much where we're focusing right now, federally qualified health centers, they saw a 200% increase through the COVID pandemic of compliance with the diabetic eye exam from 12% to 42.6%. That is being published, but already we have seen the numbers increase beyond that, and that's of course again protective of their eyesight and making sure that they don't suffer from this terrible diabetes complication if we can avoid it. I thank you very much and look forward to the panel discussion.
Thank you so much, Dr. Abramoff, for showing us how you developed this ethical AI system for diabetic retinopathy screening. We'll now move on to Dr. Pierce Keene, who is a specialist in AI and well-known. He'll be speaking on the Moorfields DeepMind collaboration, going from code to clinic. Dr. Keene is Professor of Artificial Medical Intelligence at the University College in London. Pierce?
Thank you, Jenny, for the nice introduction. If anyone would like to get in contact with me, I've got my Twitter handle in the bottom right there, and on the last slide, I'll give my email address. So I'm going to talk a little bit about going from code to clinic. So as Dr. Lim has already mentioned, I'm a consultant ophthalmologist at Moorfields Eye Hospital in London, where I'm a retina specialist. I'm also a professor of artificial medical intelligence at University College London. And although I'm not a computer scientist, not an engineer, I lead this multidisciplinary research group that aims to develop and apply AI in healthcare using ophthalmology as an exemplar for other medical specialties. I'm fortunate to be funded by UK Research and Innovation, and these are my financial disclosures. So in particular, I've previously acted as a consultant for the artificial intelligence company DeepMind. So really, I just want to update you on, what the current status of the collaboration between Moorfields and DeepMind is. This was a collaboration that started back in July 2016, where we shared a large amount of anonymized retrospective OCT volume scans with DeepMind to try and work together to develop a deep learning system for the triage and quantification of macular OCT scans. Now, let me just show you a few examples of the artificial intelligence system in action. The first is a very simple one. So this is a patient with diabetic eye disease. I believe it's a patient in their 40s with very poorly controlled type 2 diabetes. And essentially, we take an OCT volume scan, which you can see in the bottom row. We feed it into the first of two neural networks. And so the first of these is a segmentation network. So that delineates between 10 and 15 different anatomic parameters on the OCT volume. On the second row, it gives a heat map. So in this case, it's showing an intraretinal fluid map. And on the top left row, we can see the outputs of a second neural network, which is a classification network. And this gives a triage decision. So it says, in this case, semi-urgent referral to a retina specialist, and it gives a diagnosis probability, in this case, saying macular retinal edema. Now, the nice thing about this algorithm is that it can work across a range of complex macular disease. And so for the retina specialists listening, this is one that I like. If you look in the bottom row here, you can see we feed in a macular OCT scan. Coming up from the choroid, you're going to see some red there, and this is a fibrovascular PED. And so actually on the third row, in reddish orange, you can see that there's this fibrovascular PED outlined en face just above the fovea of this eye. And then if you look directly above that on the second row, you can see in blue a subretinal fluid, a trough of subretinal fluid going down from the fibrovascular PED. On the top right, you can see the posterior hyaloid, which has been delineated, which is partially detached in this case. So just to say that this has applications in vitreoretinal surgical conditions also. And then ultimately on the top left, we see the referral suggestion. So it says urgent referral, gives a diagnosis probability of diagnosing CNV and central serous retinopathy. And so it does multi-class classification. The reason I like this is because I think a lot of ophthalmologists and even a lot of retina specialists would have diagnosed this case as being wet macular degeneration. But in fact, this is a case of central serous retinopathy with secondary CNV. And although the algorithm hasn't really been trained on many examples of that, it's nailed the correct diagnosis. Now, a lot of the AI systems that are available have really been focusing on intraretinal fluid and subretinal fluid, and to a certain extent PED. But the nice thing about the algorithm that we've been working on is that it also does a range of other things. And so it segments different types of PED, serous, fibrovascular, drusenoid PED, segments the retinal pigment epithelium. So it lets us look at patients with, for example, geographic atrophy. And so this is a case where you can see on the third row on the left where there's the area of geographic atrophy. Just beside it in reddish orange, we've got lots and lots of drusen around that area. And then on the top row, it's recommending routine referral because it's diagnosing geographic atrophy in this case. One of the other things to point out about our model is that because we have this segmentation network followed by a classification network, it works very nicely across multiple different OCT devices. So including from the main, some of the main manufacturers such as Topcon and Zeiss and Heidelberg, and can easily be updated as new models of these systems become available. Now, so the next question is, where are we in 2022 with this system? Because I'm sure that there'll be many people on the call who have some sense of deja vu, having seen some of these videos before. And so I wanted to tell you a little anecdote about something that happened last year with a guy called Ken Cukier. And Ken is a senior editor at The Economist. I happen to know Ken because he wrote about the work between Moorfields and DeepMind in The Economist. And so in March of last year, he actually developed sudden vision loss. And he rang me up in a panic and he said, you know, it's the middle of a lockdown, what should I do? I was able to give him advice, get him to go into Moorfields emergency department, get seen and get treated, etc. Now, one of the things that he subsequently told me is that when he went in, at every step of the way, he was asking the nurses, he was asking the doctors, he was asking people in the surgical OR, you know, where's the AI system that is meant to be diagnosing me? And so a few days after he came out of Moorfields, he sent this tweet where he kind of gently teased us, where he said, AI is a fraud. I went to Moorfields for emergency eye surgery, and instead of DeepMind's cool algorithms diagnosing me and robot surgeons, I had to accept the excellent skill, thoughtful care of human medical staff. What a disappointment. And then he says, listen at, and then he gives the link. So it turns out that the condition he had was not something that you diagnose with an OCT, and therefore our AI wouldn't have helped. But nonetheless, the fact of the matter is in 2022, we're not actually using this routinely in patients at Moorfields. So he actually invited me on this podcast to talk about this journey that we were on. And so there's a lot of different reasons for that. But I guess one of the main reasons is that we have to bridge this AI chasm, which is to say that it's one thing to kind of, hack together a piece of code that you use to report some results in a research article, and it's a very different thing to have something that can actually be deployed at scale with the appropriate regulatory approvals. So we're working on that. And one component of that is trying to make sure that we have the segmentation validated properly. And so in the middle of last year, we published an article in JAMA Ophthalmology where we actually got about 173 patients with 2 different diseases, wet AMD and diabetic macular edema, 2 different devices. And we took data from 3 different time points. We deliberately took really morphologically challenging cases. And essentially we did the full range of disease severity, and we tested, we did really good reference standard grading for those tests. In fact, we spent up to 50 hours doing manual segmentation of just one OCT volume in many cases to try and get the best possible reference standard. Another interesting thing that we did was, in addition to doing quantitative evaluation, we did qualitative assessments. So we showed human experts the raw OCT scans, we showed reading center segmentations, and we showed an AI segmentation. And we didn't tell them which was which, but we asked them to say which one they preferred. We asked them to give Likert ratings, etc. What was interesting was that there was a mismatch in many cases between the scans that had the best quantitative scores, DICE scores, and tissue volumes, etc., with the ones that qualitatively had the best scores. So it just points out the complexity of this. The other thing that we've been doing is a global validation study. So we want to make sure that the algorithm that we've trained on data from more fields, albeit very diverse data, also works on scans from patients in Ghana, in Brazil, in Mexico, in Detroit, Michigan, and other countries and regions around the world. And so it's been brilliant to work with Eddie Korat, who will be speaking next on doing that. And we hope to publish the results of that in the coming year. And we think that there's a real opportunity here, not just for doing better segmentation of fluid, but actually bringing world-class expertise out of specialized centers into the community and into the homes of patients in the future. And so in the UK now, most optometrists in local communities have OCT scans. So the biggest chains like Specsavers, Vision Express, Costco, etc., do OCT scans as part of the routine examination. The other thing to say is that in that regard, we're doing a prospective validation study of using the system for diagnostic accuracy in that much lower disease prevalence community setting. So just to finish then, I've told you a little bit about this process of going from code to clinic. I've emphasized the clinical validation. I haven't had time, and maybe we'll come up in the panel discussion to talk about all the other things that we need to do. Really robust scoping of the use case requirements, really looking under the hood on the technical requirements, looking at the different regulatory requirements, and also looking at the different approaches to partnership with the next stages of this algorithm. And so then, as promised, that's my email address. Thank you very much.
That was wonderful. Thank you so much, Pierce, for describing how you go from the code to the clinic and showing us all the meticulous work to validate all of this algorithms. Our next speaker is Dr. Edward Korat, who will be speaking on clinician-driven AI, resources enabling democratization. Dr. Korat is a Retina Fellow, Surgical Fellow, Clinical Scholar, and he's a Stanford University Honorary Clinical Research Fellow, previously at Moorfields Eye Hospital. Edward?
Thank you for the kind introduction, and I'm honored to be speaking right after one of my main mentors, Pierce Keen. So great shout out in your talk as well. Thank you. So I'm going to be speaking on enabling the democratization of AI, about clinicians driving algorithm design. And more so, we'll be talking about doing this without coding, without private data sets, and without the use of expensive labeling. So as was mentioned, I'm currently a fellow at Stanford and soon to be in 2 weeks incoming clinical adjunct professor and honorary clinical research fellow at Moorfields Eye Hospital under Pierce Keen. So my disclosures. So my mission is to democratize AI for clinicians and researchers. And as you guys are well aware of, around 90+% of the current work in AI stays in the research phase. And I think one of the main reasons for the lack of implementation is that clinicians are not driving the actual design of these algorithms. And so there's a lot of barriers to the development of AI. A lot of these are coming down actually, but two of the main ones are computing power. You need pretty specialized computing power that requires a lot of power and expertise to actually run, and is sometimes expensive if you have it on premises. And that expertise is clustered in developed countries as well. And as we all know, machine learning is pretty iterative and complex. So from ideation to data collection to labeling the data, training the model, and then after implementation, monitoring the model and iterating, it takes a lot of work. And so if we go into our classical supervised learning archetype, as you know, it goes from input images, applying labels, which is frequently costly and takes a long time, performing a test train split, training the model, and then the model will classify different diseases. So this is a familiar slide, was in the last talk, the AI chasm. So with all of these barriers, I think that the AI chasm is very real. And it's definitely multifactorial, but I think one of the main reasons is that model design is not clinician driven. So if we instead talk about a use case first approach, where we solve the real problems in the clinic that are most relevant to our patients, I think that will go a long way to bridging this chasm. So what is clinician-driven AI? I believe clinicians, like I said, are the use case experts. And so if we design models with patient-relevant needs and endpoints in mind, which then drive labeling and model design, I think that'll go a lot further than how we currently design models. And we actually understand the pain points in the clinic more as clinicians, and there's a lot of lower hanging fruit for data integration that can be presented to clinicians to allow them to spend more time with the patient. And so the thought is, if you address a real need, this will naturally drive implementation. So for example, no one needed to be pushed to use iPhones. So currently, clinician-driven machine learning faces several obstacles. As I mentioned, there's limited access to coding expertise, there's limited computing resources, data set scarcity, and the scarcity of high-quality expert labels. And so how do we address these? We actually saw this article in 2017 in the New York Times about AI that can build AI. And this specifically referred to something called AutoML. And AutoML is a way that algorithms can be designed without the use of coding. So taking one step more to democratize access. And specifically, AutoML consists of, in its current phase, dataset management and labeling, neural architecture search, which is actually the model selection, which is automated in these platforms, the hyperparameter tuning, so the training and optimization of the network, and then the model implementation, which is frequently done with a cloud-hosted API, which is pretty standardized. And I just want to emphasize that most platforms do not require coding. And so our group first published on a proof of concept work in Lancet Digital Health, where we trained in an automated fashion without coding a few models of different modalities. And those included chest x-rays, dermatological, and OCT. And all of these models performed really well with AUCs of over 0.9. So in further work, we had a follow-up paper in Nature Machine Intelligence, where we comprehensively evaluated both the features and the performance of 6 different publicly available platforms on OCT and fundus datasets, which were publicly available. And each of these, and so in part of our feature set evaluation, and you can see the different platforms in the top row, which included Amazon, Apple, Google, and Microsoft. We evaluated if these platforms had the requisite features for high quality model designs, specifically batch prediction, which enables external validation. Long story short, but Google was the only platform that contained all of these necessary features. And so for our actual performance evaluation, and you can see in the left-hand panel, these models broken down by data set, and each of these points is a separate trained model. We can see that they all generally perform well in their F1 scores. And we can also see that OCT models perform better than fundus models. So this is probably due to the lower dimensionality of OCTs, them being non-color and things like that. And then on the right side panel, the same performance is broken down by platform. And here we see that Amazon, Google, and Microsoft are generally all performing very well. And each of these points is a model trained without coding. And here's some precision recall curves demonstrating the same. Most of these models performing really, really well, especially Google design models and Amazon. And so one of the other main things that we wanted to address was looking at the saliency maps and to try to address explainability. And while there are a lot of issues with these kind of maps and a lot of pitfalls, this is a place to start. And so in a more inherently opaque platform, such as AutoML, where you don't pick the actual features and things like that, we thought that this was important. So our follow-up work on this, we decided to evaluate the use of region-based attribution maps on something that clinicians actually cannot do. So how cool would it be if we can design a model that predicted sex from retinal fundus photographs using AutoML without coding, and then looked at the region-based attribution maps to see where were the salient regions in the fundus photograph for that decision. And we found that the vessels, the macula, and the nerve were actually all important features, and especially interesting in column 2, where images that were deemed ungradable by humans actually had salient regions and correct predictions by the model. And so this work was actually replication of a famous work published prior in Nature Biomedical Engineering by Google. And this was with expert coders at Google with all their resources, and their AUC was 0.97. And our AUC without coding with publicly available data sets was 0.93, so pretty close. So fortunately, a lot of the UIs of these platforms are pretty simple and mostly drag and drop. And it's a three-step approach where you upload the data, visualize the data set, and perform the labeling, and then train the model and perform evaluation. So now going back to this slide on the obstacles to clinician-driven ML, we've shown how the lack of coding expertise and computing resources is mitigated by AutoML. And how about data set scarcity and the lack of high quality labels? So our group then performed extensive evaluation of publicly available ophthalmic data sets. And we published this in the Lancet Digital Health. And there's actually 94 open access data sets available with over 500,000 images from over 100,000 patients. And we put all of these in a compendium that can be easily accessed on idatasets.com for anyone to use. So the question remains, can you build a high performing model without coding, without computing resources on premises, without your own data, and without expensive expert labeling? So let's go to the labeling component. In follow-up work, which is unpublished at the moment, we utilize this framework of self-training to address label scarcity. So what we did on the left side, you can see we first trained what's called the teacher model, where we used high quality labels, which were publicly available, to train a small, well-performing model. We then used that model to label a large unlabeled data set. And then those generated labels and images were integrated back into an additional round of training, and this was termed the student model. And the results here show that the student model performs better than the teacher model without any additional labeling effort, and both actually perform better than the bespoke model created by coders, which is in green. So we actually performed a limited evaluation against diabetic retinopathy referral model, which is IDX, which was, as you guys know, FDA approved. And in our evaluation, AutoML matched IDX performance. And this was without coding and without labeling. So I think I'm out of time. I'll skip through a couple slides. There are a couple low-hanging fruit for use cases which do not require FDA approval: collation of data, clinical trial recruitment. We always caution against the use of these without proper approval, and don't use patient data first. And robust failure mode interrogation is very important. There's hidden stratification that can happen in the real world, and auto distribution performance is frequently not as expected. And I hope I've showed how we can address all of the obstacles to clinician-driven ML. And so this is a graph on the left side where the efficiency of transport is shown against body weight. And a human on a bicycle is actually the most efficient form of transport. And that shows how a simple tool can unlock human potential. And so when Steve Jobs introduced the PC with the first Apple, he mentioned that he thought this was a personal computer, or that this was a bicycle for the mind. And I hope that AutoML will be a bicycle for the clinician to unlock our potential. And so these are my references. And if not you, who? You should be driving algorithm design. You know the pain points of our patients in the clinic. And I hope this will help bridge the AI chasm. Thank you very much.
Thank you so much, Edward. That's fascinating. Let's go ahead and go to our panel discussion. And I'd like to start with a question directed to Dr. Abramoff. Edward showed beautifully that his self-designed, auto ML program performed as well as the IDXDR. Any comments on that?
So evaluation is more than just testing against some data set which has been labeled in some way. I mean, what is interesting is there racial bias? Is there, other biases? Can you exclude that? Do you know about diabetic macular edema? There's a lot of truth that goes into evaluating an algorithm. It's cool that you can match the performance. I think, the challenge is not anymore how high can your AUC curve be, but more how do you implement it? How do you know it's safe? How do you make sure that the users know what to do, that you, in fact, in a clinical setting, which is not ophthalmology, I mean, right? Much of what the previous speaker, I forgot his name, was discussing was about essentially physician or ophthalmology driven AI, but that is very applicable to when we use it in clinic ourselves as a retina specialist or ophthalmologist. Most of the AI where I see it happening is in the front lines of care, where there's no such expertise, and it's a very different environment. And so while this is useful for, if you want to use it on your own patients, and you can, right? There's no FDA regulatory framework around it. You can do so, but if you want to use it in a very different environment where people don't know what it actually does, I think that's more risky. I'm glad to say that the Joint Commission is working with me now to oversee, or better oversee, those AIs that don't go through FDA regulatory approval in the US at least. But yeah, so there's a lot more to be said about this.
Right, that it's not being used autonomously. I think, Pierce, you have a comment?
Yeah, no, I just wanted to highlight that with this kind of code-free deep learning that, well, first of all, I would say it's clear from all the talks that even for bespoke models created by the best software engineers in the world, that really rigorous validation and care is required. And that would be even double the case if you're talking about something generated by a clinician. So I don't really see these necessarily being used for direct clinical care anytime soon, even if there's some loopholes with this. But I kind of imagine it, it's like if clinicians have, imagine if we have like 100 ideas and we could do the early kind of early proof of concept, and maybe out of those 100 ideas, like 10 of them, or maybe even just one of them, shows some signal that it should be made into something bigger. And then that is at the point where you could then engage with the computer scientists and the software engineers and the commercial partners who really know what they're doing in terms of the validation and the regulatory approval and all of those things.
Thank you both for clarifying. So I have a question. We've worked in our system on an AI for sickle cell retinopathy, say. And then to get it to the next stage, we would need to validate, right, the system using multiple images from multiple centers. Now, Dr. Cheng had touched upon this, and Pierce and Jayashri, you also, and Luis, that different systems may not work, right, because of the way the images are labeled and the way they're saved. So my question is, using your data bank that you used in Moorfields, how did you get all these different images from different places to be actually able to be used? And what recommendations do you have for people who want to do the same thing?
Well, so that's kind of a complicated question. It's a good question. So I think one of the advantages that we have in Moorfields is that we have large amounts of data from all of the major imaging vendors. So just in Moorfields, we have 30 Cirrus OCTs from Zeiss, for example, and we have probably similar numbers from Topcon and Heidelberg. And so we had an advantage to be able to do that. I think then the second point is the way that we've set up the algorithm I displayed is this kind of it's not an end-to-end approach. It's this segmentation network feeding into a classification network. So if we train both of those on images from vendor A, and then we want to then repurpose it for vendor B, then we can essentially, what we showed in our paper was that we can retrain on a small number of images, retrain the segmentation network on a small number of images from vendor B, keep the same classification network, and it works very nicely on the new device. And so that was our approach, but there's lots of other ways.
So kind of just fine tuning it, if you will. Dr. Abramoff?
No, I want to make sure Piers finished, because there's another way of doing this. I mean, this is a problem where you need to normalize across different sites, which is what Piers is describing. If you run, no, you cannot do that. But if you use biomarkers like hemorrhages and exudates, they are present in all these images, right? Otherwise, clinicians would not be able to diagnose the disease. So if you rather make your AI around biomarkers and detectors for those, you get rid of a lot of these normalization problems.
I see. So you're basically identifying the biomarkers, almost like a machine learning sort of approach, as opposed to deep learning.
Well, it's still deep learning because the detectors are typically trained using deep learning, but it's a detector, so you know what it does. You know, oh, this detector is for large hemorrhages.
Right,
this
you identify.
Small exudates. This detector is for SursiNet exudates, et cetera, et cetera. I mean, Piers and Keen have been, discussing this a lot, and it's always fun. But I think the more you take the image as you don't make any assumptions about what is in the image, the more you need to normalize and do a lot of effort on that.
And so I totally agree with both, and I just want to add a little bit on the validation piece. So extensive validation, I think, is the most important thing. I think it's more important in building trust than explainability, quote unquote. And yes, if you have a segmentation network that then feeds into a classification network like the DeepMind algorithm, that helps because you can see the underlying segmentations. But I think even the current way that the FDA does approvals with a pivotal trial, which is potentially disease enriched, does not address what we'll see in the real world. So I think we need real world validation at multiple centers, potentially federated, as well as continuous monitoring. So you could have a subtle pixel intensity shift by an update that's put through by Heidelberg, where a pixel can shift by 1 or 2 in luminance and no one would know, an engineer doesn't see, and that could change your outputs. And so unless you're doing monitoring that's standardized, that's not going to be picked up for out of distribution shift. So I don't think that the current way that the FDA does it is even adequate.
Well,
sorry.
Can I push back on that? So you just mentioned that it's important that clinicians become closer to this. The more hurdles you create, because there's a way to solve this, right? Once you have implemented it and the FDA is requiring that, there's continuous efficacy monitoring, meaning you continue while it's in the field to monitor its performance, which I think is essential to continue trust. But we had to spend almost 20 pivotal trial of 900 patients, right? And so if you make the burden even higher and say, well, we did 10 sites, right? But if you say, well, we need to do this across the world with tens of thousands of patients, there's only 1 or 2 companies in the world who can do this. And I don't think that was the intent of your presentation, where you said, well, rather we want to bring it closer to what clinicians are doing.
Great. I'd like to move on now to a slightly different topic, and that is the uncertainty. Luis, you showed it very nicely that, machine can sometimes make the wrong diagnosis, right? If it doesn't know what it doesn't know. So that's always a dangerous thing when we're training our residents and, for doctors, like if you don't know what you don't know, which is kind of like, non-intuitive, then you could make this big mistake. So do you want to comment a little bit more on how can we know what our AI systems don't know? And is it just really being careful about the inputs and then following what the outputs are very carefully?
Yeah, I think that like I was saying in my presentation, we need to have a way for the algorithms itself or whatever we develop to say like this image has never been encountered. This is something that I don't know how to deal with. And like you said, it's not about just teaching the algorithm something that has never seen, because that's basically impossible. But it's just trying to figure out what is implicitly in the model. And it's a little bit tricky because I almost giving up on explainability. I just try to explain something that comes from the models, and it's, I don't know, everybody just picks their images and say, oh, I see something here, but that's just the big image that they get. In theory, they are going to have, like, these models have more neurons that we're going to have in our brain. So explaining them is going to be impossible. It's just impossible for us to hold. We can set key points. I like the idea that Pierce is saying, that he's not training the model directly to go from here's my image, here's my diagnosis, but to first find some features, find some particular segmentation of different diseases, and then get the diagnosis from there. So maybe establishing those key points along the model that we can teach more individual steps to get to the final step, and then make sure that each individual step makes sense and each individual step is provided without uncertainty, will give us a good solution for that. Yeah, and at the end of the day, it's also, it makes more sense to teach the model more things than less things. I know that these models work really, really well for a simple task, but nobody just knows how to do just one simple thing. They will perform that simple task better the more things that they know about other tasks at the end of the day.
Thank you. So perhaps we'd have to have in there, the different features that are looked for, and then there might be a category unknown, not seen, perhaps, not detected. So Dr. Kalpathy-Kramer, in radiology, when you use these images, you showed us that picture of the breast tissue and, trying to diagnose the dense versus the fatty breast tissue there. What takeaways can you move from radiology into our field to help inform us about different things such as image texture and the importance of that in terms of analysis?
I think, pretty much everything we've seen in radiology, we also see in ophthalmology, and the things that we've talked about. So one is portability of the algorithm across devices. We see that in many different domains, and we see it here. And things such as Pierce mentioned in terms of sort of decoupling different intermediate steps and adding maybe a segmentation step that's device specific, and a classification step is one way to get around that. The concern I still have is that you might get it working for 2 or 3 devices when you add a 4th one or a 5th one, and unless you're continually monitoring it, you just don't know that it's working. There's a lot of work in the computer science field about the use of out of distribution metrics. And the idea is, if you have a good metric to say how far away this particular instance is from your training data, maybe you can decide if it should be used on that image or not. Unfortunately, it's still a work in progress. There's a lot of research happening there, and I think something we have to continue to, there's whole workshops devoted to that at these computer science meetings, is how do we come up with a good metric of out of distribution that translates to a likelihood of whether the algorithm is going to work on that particular case or not. Uncertainty is another really good example. We've done a ton of work in uncertainty, and there are many different ways of estimating uncertainty. And what we've shown is that uncertainty can be sort of a proxy for performance. So if you train your algorithms well, you can identify sort of gray zone images or things on the boundary between classes where the decision boundary is likely to be uncertain and therefore potentially wrong either way. And if you don't do that and you actually force your algorithm to make a decision, you get algorithms that are very brittle at the class boundary, and they'll sort of flip-flop between positive and negative for sort of uncertain cases. And we have to be quite careful about how we do that in this domain as well. Repeatability is another example we've seen where, again, we see this flip-flopping. So a person is classified as positive in one thing in a slightly different image, and they flip negative. And we've seen some talk of that here, but I think quantifying how repeatable the algorithm is and reproducible it is important. And bias and fairness are, again, big issues and things, just making sure. So same lessons, same challenges from radiology as well.
Right. I think, it's one of the challenges in ophthalmology. We like to use a lot of the clinical trial data. And as we all know, the clinical trial data is not very heterogeneous. It's very homogeneous. And so then to apply the data, to apply the AI that was trained on this data, say to different ethnic groups, socioeconomic groups, is a little bit of a leap. And so there is that chasm. I love that slide that shows the chasm from, getting the algorithm into clinical applicability that I think truly exists. But I think with all of you collaborating on this, I think that chasm is going to be broached in a good way, and that we're going to be able to get to an AI system that is clinically useful and meaningful. So I thank you all for your outstanding presentations and your excellent comments. We're going to go now to a break from our sponsors, Apellis and Notal Vision, who are our silver sponsors. There'll be a short video that plays, and immediately afterwards, we'll resume with session 5 with Dr. Ted Lang. Thanks for joining us.